at numerous national and international platforms. He is the recipient of Organization for Pharmaceutical Producers of India Scientist Award, Shakuntala Amirchand Prize, and Dr. D.N. Prasad Memorial Oration Award of Indian Council of Medical Research. There are many more awards and rewards to his name. I'll fall short of words. So without any further ado, I'd like to hand over the dice to our speaker, Dr. S.S. Sharma, Naipur Mohali. Thank you.
गुड मॉर्निंग आवाज़ नहीं आ रहा स्टूडेंट्स का गुड मॉर्निंग yes yes thank you very much actually that one okay I think already the stage had been set yesterday one of my guru actually Professor Y.K. Gupta he inaugurated the conference over here actually and I'm lucky actually and I thank Dr. Sumit Gupta for inviting me over here actually for this wonderful morning and we had actually already breakfast with sunny day and all. I think it can't be better than this kind of a day. Like moving to my talk actually which is on drug discovery and development of CNS drug actually. Why I just thought of like selecting this topic regarding CNS drug because I want to let you aware about what kind of work is going on in the CNS drug discovery area and how you can enter into the research. So I'm going to share like some of our data we have generated related with the CNS drug. If you look at kind of a like requirement, why there is a need of CNS drug. Now it has been mentioned that 1 billion people suffer with neurological disorder. And this number have increased actually after the COVID-19 pandemic because number are increasing like anything. Some of the common disease condition if you can see in the list actually starting from like epilepsy to migraine to stroke, there are several disease conditions in addition to neurodegenerative conditions like Alzheimer's disease and Parkinson's disease. The number of these neurodegenerative conditions are basically increasing. So that is why there is always a need of new pharmacological interventions which can be targeted for CNS disease conditions. If you look at actually like the global CNS market, like US $110 billion it was in 2022 actually, and this is going to increase to around $238 billion with that. So this is a huge increase in this like kind of a CNS market for and the requirement for CNS drug. And they are considered to be the 5th of the sales of the blockbuster drug after like those hypolipidemics or cardiovascular drug. But again actually there is a issue of like always the high attrition rate associated with the CNS drug discovery. Since like most of you are pharmacy students, you must know like how a drug is being discovered. So when it's come to CNS drug, like I think first you need to identify the target, what is the best target or ideal target for CNS disease conditions actually. And then you have to, we have like a lot of very good chemists, natural product chemists actually, they can synthesize or they can generate some molecules actually which can be looked into generating as a new lead. So once you are generating a lead actually that can be used, it is displayed, yeah, okay. Okay, so now you have to generate a lead compounds actually and then you need to modify that lead compound so that you have a drug actually which can cross blood-brain barrier because this is one of the major challenge actually for any CNS drug for reaching to the market because most of the drug get failed because they are not able to cross the blood-brain barrier. Then as a pharmacologist you can have the in vitro model and in vivo model for discovering CNS drug and then the clinical phase of drug discovery and development will start. Generally like when we talk about $1 billion for discovery of the new one drug actually 70% goes to clinical phase and 30%, about 30% goes to your preclinical or the discovery phase. If you can look at the top selling CNS drug, you will see most of the drug actually related with the antipsychotics actually or antidepressants actually, most of the drugs, because they are being used for treatment of, like if you take olanzapine is antipsychotics actually, then paroxetine and all, these are antidepressants, they are in the top list. But if you see that recently, like I think 2022 data from US, you will see that pregabalin, DMF actually, there are several new drugs which are coming to the market. Even last year also there was a drug actually, like one of the monoclonal antibody which was approved for Alzheimer's disease actually, and that is already doing wonder in US market. But always the issue is that 85% of investigational compound never see the pharmacy shelf. So now, like, this is one of the challenge actually, and what are the challenge can be there for CNS drug discovery is like complexity of the brain. Our brain is a highly complex organ. If you know, we have like billions of neurons and actually 10 times of them supporting cell like astrocytes, glial cells, and oligodendrocytes. You have actually all these cells in the brain actually, and that makes brain is one of the highly complex organ. And the plus the 20% of the oxygen requirement, okay, and 50% of like total like your energy requirement. That's like, if you consider brain, there are a lot of requirements are there, and that's why it makes brain as one of the complex organ. Blood-brain barrier, there is a challenge always there, and the efflux transporter, if you are taking a drug, that drug would be efflux out actually. So we need to have a kind of a strategy where you can combine your drug with the efflux inhibitors. Patient heterogeneity, like response of the patients vary from one person to one person. Like if you see this Tacrane initially when it was launched, 30% of the patients were respondent, others were not actually. We have to look into like pharmacogenetic variation, why like some patients are responding, why some are not responding. There are like multiple target of every disease condition, because pathophysiology, either etiology not completely known, even if it is known, there are multiple targets. We have to target those therapeutics. One will not help actually. You have to investigate neuroprotective potential in number of disease models, then only you have to take that particular molecule for further development. Otherwise, the chances of the failures are going to be more. Predictive validity of experimental efficacy data is another challenge, and another biomarker. Like we have to have a like biomarker drug discovery, whether it's stroke biomarkers, Alzheimer's or Parkinson. This biomarker discovery is required. And abuse liability. One of the always the challenges that abuse liability. If a drug is causing abuse, liability or dependence, then you cannot take that drug to the market, because there would be huge issue, and US FDA or Indian FDA would not approve these kind of interventions. So now you can see the brain health post-COVID-19. You can see there is a brain stroke is there, neuroinflammation is there, depression can be there, anxiety can be there actually, and cognitive impairment can be there, many more. So now you can think actually like brain health after COVID-19 have changed drastically, and there are a lot of cases have been reported. Since like I cannot discuss like all of these actually, I'm just going to take up this brain stroke today in some more detail. If you know that like brain stroke, basically there are two types of stroke. One is ischemic stroke actually, which involve about like 80 to 85% of patient, and there is a hemorrhagic stroke involving around 15% of the patient. In case of the ischemic stroke, there is like one of the major artery in the brain which is get blocked actually, which we call middle cerebral artery. If you know about the blood supply to the brain, like we have like common carotid artery, like two common carotid artery and two vertebral artery, they enter into the brain actually and form a circle of Willis actually. If these arteries are get blocked, actually that can lead to the reduction in the blood supply to the brain, and that can cause a stroke. And most of the cases, like 80 to 85% of the cases are related with the ischemic stroke that has been shown. There, like if you look into the global prevalence, actually it used to be the third leading cause of the death and the first major cause of disability. So most of the stroke patients, actually they will get half of the body get paralyzed in those patients. It's not like that our body will not give you the warning symptom. Always our body can give you the warning symptom by inducing like TIA, transient ischemic attack. So some people, they feel like blurred vision or weakness in their limbs and all. That can be the indication of stroke. So in future, actually, these patients actually might develop a stroke, full-blown stroke. So there are, like, if you see the prevalence, like around like 0.69, like around 0.69 million death has been reported, and prevalence 1.29 million cases are there, like every year you can find. And these are the word actually all of you need to remember, like FAST, F-A-S-T. So it's a common, I think if you remember this like face, they will be drooping of one side of the face. You can see in the slide, like one side of the face of that female is getting droop actually. That indicate actually change in the expression of the face and the drooping of one side of the lips actually. Smile, if you ask a person to smile, they were not able to. Then arm actually, like weakness in the arm. You can ask a patient actually to raise their arm actually. If there is a weakness, actually one arm will come down. Okay, that indicates there is a weakness, or maybe like there may be half of the body get paralyzed and the speech, slurred speech. So generally, like there is a slurred speech, like not clear, it will not be very clear. And T stand for time to act. So this is an emergency, stroke is an emergency, and immediately you have to take that patient to the hospital, and most of the hospital like PGI, AIMS, or nearby hospital, even your MMU hospital, medical college, actually they also have like kind of a stroke unit for the treatment. 4 hours or maybe 48 hours. So a lot of research is going to be done actually, where we can develop neuroprotective agents. These two thrombolytic therapy, like you would be knowing about recombinant tissue plasminogen activator. They are being used. This LT-plage and 10-actiplage, they have already been used. Actually, they are in use for clinical use for opening this blockade actually, so that the blood flow can be restored. You can see over here actually in this particular slide, this dark portion is basically infected portion, and surrounding this portion we call this penumbra. The penumbra is the area actually in which actually if you restore the blood flow, then damage, neural damage can be prevented. This is the advantage. So that is why, like after giving thrombolytic agents, the blood flow have been increased, and that area will not develop into the necrosis. Otherwise, this necrotic area will keep on increasing with this actually. There are several pathways which have been investigated for stroke, like glutamate release and glutamate receptor activation has been looked into, so that you can block excitotoxicity, inflammation can be blocked, calcium influx has been blocked, necrosis, apoptosis, like by developing anti-apoptotic drug, then antioxidants by targeting oxidative stress, neuroinflammation, neuromodulation, and regenerative therapy. So there are chances and there is a scope. As I mentioned, like there is an opportunity to look for and developing newer pharmacological interventions which can be used for treatment of a stroke. There are more than 100 clinical trials actually have already gone actually into the picture, like phase 1, phase 2, phase 3 clinical trial for developing neuroprotective agent. But most of the time actually these interventions have failed in clinical stage, and that's why like till date actually we do not have like good neuroprotective agents which can take care of the stroke after 4.5 hours. You take nimodipine, which is a calcium channel blocker, which was also, because it's a cerebro selective calcium channel blocker that was used for that, and many more agents actually acting on NMDA receptor or excitotoxicity they were developed. So there is always a kind of a translational challenges from bench to bedside, and what you can see, like these are the challenges, and as a postgraduate student, those who would like to work on for their masters or PhD can try to take up these challenges actually, how you can take up these challenges, like target validation, biomarker discovery, experimental model, can you develop newer model, clinical development, ethical challenges are there because already patient is in the hospital ICU, and whether like their relative give you the permission to go for the new clinical trial or not actually on that. So what is very important is like target validation. Whenever you are selecting a target, you have to look into whether that particular target is there in the human or not. Most of the time we use the animal or we use the in vitro models actually, we have to see that that particular cell line, if do not have that particular target, you need to transfect that cell line so that you can investigate various uh, pharmacological agents. And you have to see whether your drug is reaching at the site of action. That is another point that you have to take into the consideration. And if you see this particular slide, like it's not only just neuron is a target, your other supporting cell can also be target like microglia, oligodendrocytes, then pericyte, then you have like EC space. There are several targets which had been looked into. Only you have to look into like which one is the basically the ideal target for this one. So there are multiple targets which had been shown in this particular picture actually, and these interventions can be looked into these kind of targets. There are challenges with the stroke model. Okay, the model what we are using actually like it should depict kind of a cerebral infarction which is happening in case of like human. So that is why we are looking into like developing newer model actually for stroke, whether it's in vitro, in vivo model. In vitro you can take a primary neuronal culture and you can do, or either you can take SHS55 cell line actually and you can expose that with excitotoxic agent to check the neuroprotective potential. If you look at like stroke model, like we have like in vitro, in vivo stroke model, and one of the commonly used stroke model like global cerebral ischemia or the focal cerebral ischemia. Focal cerebral ischemia model directly indicate the stroke model because in that we are going to block middle cerebral artery because this is the artery, major artery which is getting blocked in case of stroke patient. So if you can block this artery, then you can produce the symptoms, whatever the similar kind of symptom like fast which are happening in human. So that we can do actually. So this is the model we have developed actually, and we are using this model, middle cerebral artery occlusion model. In this model, what we are doing actually, from external carotid artery, we passing a filament actually through internal carotid artery, and then we are extending this filament up to the middle cerebral artery. And at the point of middle cerebral artery, then we record the blood flow. So whenever we have a laser Doppler blood flow monitor and we record the flow, the moment blood flow comes down to around 80% actually, we think actually, okay, there is already ischemic damage has happened. So we go for like around 2 hours of occlusion actually, and then reperfuse it, and then look for developing few interventions. If you look at the blood supply to the brain, and this area basically, like if you can see, this area is basically supplied with the middle cerebral artery, and that is why like you need to concentrate on that. So there are like various parameters we need to monitor. We monitor blood gases, we monitor like blood pressure changes and all actually to check whether the surgery is perfect or not. And the blood flow, cerebral blood flow, the area which is supplied using the laser Doppler blood flow monitor, to some of the instrumentation shown over here. After this actually, like we take this TTC staining and we sacrifice the animal, we make it a coronal section, and then we go for staining. And this stain actually basically stain only the live cell, and the dead cells will not take up TTC stain. So this is the point actually, if you see in this particular slide, there the white portion what you are looking at it actually basically that indicates there is a brain damage with this actually. Imaging technology nowadays, like many institutions like AIMS, we have used this MRI actually to check the damage to the brain using MRI, because in these cases like there is no need to sacrifice the animal for that. Then you need to go for neurological function test where you can have like various scoring system at different time interval, and then you look for this one. Another model actually which can be used is the bilateral common carotid artery occlusion model, and in this model actually like two common carotid arteries are basically like blocked actually for 30 minutes actually, and then you see the brain damage. So some of the neuron in the brain like hippocampal neuron, they are basically more vulnerable for damage. So they just get blocked actually, and that induce global cerebral ischemia model. There like we use gerbil because in gerbil basically like circle of Willis is not fully developed. So just blocking actually two common carotid artery can be sufficient. instead of like blocking all four major arteries which are supply blood to the brain. So this model is being used in the gerbil. Then what also you require, like going for comorbid condition. If you see, there are a lot of risk factor of the stroke is there, and one of the risk factor you see that if a person is having hypertension, diabetes actually, then the chances of like increase in the stroke infarctions is going to be high. So you have to have actually, or you need to use like obese model for developing this blockade, MCO blockade, hypertension or diabetes, and then you see that how much infarction have increased and check your intervention whether that is affecting that particular experimentation. All of you also, those interested to do research, they must remember this, these guidelines, and they should follow those kind of recommendations, which is stroke treatment academic industry roundtable recommendations, because these recommendations can help you to do your experimentation in such a way so that you have the better reproducibility. This is the advantage, like whatever you are doing actually it should be reproducible. Okay, otherwise there's no value actually like that. You have to take into the consideration. So what you require, the rigorous, robust and detailed preclinical evaluation in variety of models. Just one model is not sufficient like MCA. You go for global, you go for like constriction of MCA, or maybe you can go for thrombotic induced photothrombotic model of stroke, and plus like comorbid conditions you will need to take into the picture. Another point, you have to always look into like developing biomarker. Like for stroke, there are several biomarkers which are neuron specific like NSE, neuron specific enolase, then 100S, 100 beta, that's another protein, and glial fibrillary acidic protein, GFAP actually, that's also another specific. So there are some of the biomarkers you have to take into consideration. You can have like pharmacodynamic biomarker, disease-specific biomarker, as well as like pharmacokinetic and those kind of biomarker you have to look into. And the clinical development also, like as I mentioned, like it is challenging because the patients are having like a stroke and half of the body is already paralyzed. So a lot of ethical consideration, they need to be taken care actually, because most of the hospital, they already have the human ethics committee, and once like they approve the protocol actually, if it's a new drug, then you need to take permission from CDSCO for going for clinical trial. That is also you require. So we have like a stroke research laboratory which is equipped with most of the advanced level instrumentation for doing gene expression, protein expression, and other studies. And we use gaseous anesthetic system, homothermic blanket system, and many more instrumentation for doing research. So just actually like I just mentioned about some theoretical part, just I'm going to show you some of the result actually in this particular study. We have targeted peroxynitrite and PAR pathways. PARP is nothing but poly-ADP ribose polymerase pathways that we have targeted, and we have used different interventions like whether using peroxynitrite scavenger or PARP inhibitor can show some protective effect. Here like we have used peroxynitrite scavenger like FET-MPYP and FET-PPS, and several of these PAR inhibitors like kind of a this ISO, and then 3-aminobenjamide and many more actually to look into whether they are effective in not. And we also looked into like therapeutic window studies for peroxynitrite scavenger, and this work we published in the British Journal of Pharmacology, and we have done actually at 6 hours, 12 hours different time point this kind of intervention studies was done, and you can see that FET-MPYP, which is a peroxynitrite scavenger, you can see this is the kind of ischemic group, and another like group is the treatment, and you see the kind of a protection was there. And then this is the infarct volume. There is a significant reduction in the infarct volume as well as the edema was reported with that. We looked into mechanistically whether the kind of a peroxynitrite scavenger has any effect on peroxynitrite generation. That was looked into it actually. Then neurological score, as I mentioned, some of the criteria that you have to look into the paralysis in animals, and that you have to score, and that scoring was done, and you can see it with the treatment. There was a reduction in the scoring system model, and we find like and that was reversed with the treatment with peroxynitrite scavenger. neuronal density results are shown over here actually. Then DNA fragmentation was there. In addition, like when we were following these guidelines, we have looked into that diabetes actually, in diabetic animal, there is an increase in the infarction. That indicate like diabetes can exacerbate ischemic brain reperfusion injury. So we have checked actually there's significant increase in that one. And those were the guidelines that you need to do in the comorbid condition, and that's why we have got it. So we use this edaravone. Edaravone is one of the pharmacological agents already approved, even US FDA for neurodegenerative condition, or in USA also they are being, this particular agent is being used for stroke also actually. Now you can see this edaravone 1, 3 and 10 mg per kg dose showed significant reduction in the infarction for this cases. So these studies was done actually, and they are showing like very good neuroprotective potential for this kind of disease condition. So what the suggested road to translation? As I mentioned, like there are a lot of opportunities, lot of challenges are there. Challenges I discussed actually, like blood-brain barrier permeability should be there. Okay, target validation has to be there. Experimental models, multiple models need to be used and all. Several of these challenges I discussed. So this is the suggested road to translation, like from bench to bedside, what you need to do, that had been shown over here actually, how the translatability of any pharmacological agents can be good actually. So that we need to follow this particular route. My lab is working on mainly on the neuropharmacology like CNS research, but like since as an institution in NIPER, we have developed like diabetic complication laboratories, safety pharmacology laboratory, and the cardiovascular research. So where like we use this myocardial infarction model, then we use cardiac hypertrophy model. Various models have been used in our lab for investigating neuropharmacological intervention. I would like to thank my students because they were really involved in like developing the data which I presented actually. So I thank most of them. One of my student, Bhupesh, he has done a lot of work on the Parkinson disease, and he's going to Baylor School of Medicine, US actually for postdoc. So that's why like most of the students, like they are really working hard actually. And I also want like some of the students from Mulana University actually, MMU, should come to NIPER actually, but you have to be competitive so that you can get admission in NIPER for your master's or doctorate program actually. With this, I would like to thank, actually this is the beautiful campus of NIPER Mohali, and if whenever you are visiting Chandigarh or anytime, you are welcome to visit NIPER Mohali. Thank you very much. If you have any question, I would be happy to reply.
Hello. Thanks, Professor Shyam S. Sharma, for an excellent, very well illustrated presentation, and it will informative for students as well as faculty also. Any questions?
Hello, sir. Sir, I wanted to ask you a question that given the complex pathophysiology of stroke, would it be a good decision to have multiple drug therapy for its treatment?
As I mentioned actually, like it's a complex pathophysiology. So when actually you are looking into like there is a excitotoxicity involved, there is oxidative stress involved, there is the inflammation is involved. They always like there is a search for a pharmacological agent which can act on like 2-3 pathways. Okay, so there are various pharmacological interventions. They are not just acting on one. Okay, they are acting on the multiple pathway, like Trilajed was initially developed, acting on the multiple pathway. If they are not acting on multiple pathways, actually people are like taking two molecules and they are combining them with a linker to develop a molecule which will have the multifunctional activity. So there are always like a lot of molecules actually which have been looked into, but as I mentioned, actually like translational failure.
Yes.
Okay, and the reason for translational failure, as I mentioned, that most of the drugs which are discovered actually at in preclinical stage, they were not completely tested for their neuroprotective potential. Even like it has showed the 40% neuroprotection, they were taken for the clinical development. So what we need actually, a drug must show at least like 80% of the neuroprotection in variety of models. Then only it should be taken for further development for clinical stage like phase 1, phase 2 or phase 3. So that kind of strategy is going to be required.
Thank you, sir.
Good morning, sir. My name is Rahul Kumar. I am from Pharmacology Department, MMCP, MMU Mulana. So my question is that there are now the depreciating cases of HIV associated nephropathy and brain nephropathy. So, sir, we have heard about different diseases like Epstein-Barr viruses. We have a report syphilis and aortic syphilis. Sir, in that case, if a patient is already taking a lot of drugs, which is wasting their muscles and all those adipose tissues, and their lipid peroxidation levels are very low, and those adverse reactions such as a zidovudine drug has a very adverse reaction on brain, sir. Sir, in that case, how would we be able to devise a new drug delivery system so that we can remodulate the frequency of ischemic injury produced in the patient who are suffering from different venereal diseases?
Yes, I can tell you like there are several disease conditions actually like they can impair your blood-brain barrier. Okay. Like neuroinflammation, and wherever the neuroinflammation is there, the blood-brain barrier get impaired actually, and a lot of molecules which are circulating in our blood actually, they will cross the blood-brain barrier and they can start damaging the brain. So what we required actually like how we can actually prevent this kind of impairment in the blood-brain barrier. When you are saying like a lot of some of the disease condition actually where already some neuronal damage can be there, like there are several disease conditions which I mentioned to you, like whether your Alzheimer's, Parkinson, that also going to induce the neuronal degeneration and all actually. So in those cases actually we'll have to look for a kind of a combination of the therapy which can have the better. Like now you see the superoxide dismutase. It is very good antioxidant, but it cannot cross blood-brain barrier. Okay, so that is why it's better actually if you can give kind of a SOD mimetics. Okay, which are a small molecule like Tempol, which is a SOD catalyst mimetic. It can be very well given and can be cheaper than the SOD to induce antioxidant effect and which can have the better efficacy. So there are kind of a various strategy you can develop for the treatment.
Yes, sir. Thank you, sir. Sir, meanwhile, one more question.
Yeah,
Sir,
yeah.
whenever we have a blood test investigation, most of the times in this era, we have an increased level of serum albumin in our body. Whenever we go for the testing of our urine or some samples like that. And there is a very well was connection between a serum albumin rise in concentration of serum albumin and then the rise in the effective capability of dendritic cells. And that in turn can damage the neuronal pathway very much, up to 80%, sir. So in that case, what would we say, sir?
So, like when you're saying like albumin, it's not like most of the disease condition where you see the nephropathy is there.
Yeah.
Lot of the blood, urea, nitrogen, creatinine and all actually like they get increased. Even nowadays they consider like EGFR actually, like you can basically like relate with the EGFR that's used as a marker for nephropathy or so. When you're talking about the albumin actually, even actually we have published one study where the albumin has showed the neuroprotection. So you can go through that study and look at it actually. Okay, we have already published that work actually, albumin, how it can induce neuroprotection in animals actually. And there are several reports in the human also, like taking the albumin and relating it with the neuroprotection. Okay?
Thank you, sir.
Any other question? Okay. Thank you very much.
Thank you so much, sir, for delivering this informative lecture, and thank you so much to the chair session for holding the session. Now with this, I request the chair session members, Dr. Jyoti Sareen and Dr. Dinesh Kansal, to please come up on the stage and appreciate the speaker, Dr. Shyam S. Sharma. I do invite Dr. Dinesh Mehta to felicitate the chair session member, Dr. Jyoti Sareen. I request Dr. Akash Jain to please come up on the stage to felicitate Dr. Dinesh Kansal, chairperson of the lecture. Thank you, dignitaries. The next lecture of this session will be delivered by Dr. Indrapal Singh Naipar Mohali. The chair session for the lecture will be Dr. Richa Shri and Dr. Nitin Bansal. With this, I invite Dr. Nidhi to please come up on the stage and present a sapling to the speaker, Dr. Indrapal Singh. I cordially invite Dr. Neelam to present the sapling to the chairperson member, Dr. Richa Shri. I invite Dr. Seema Bansal on the stage to welcome Dr. Nitin Bansal, chairperson of the lecture. Ladies and gentlemen, I would like to give you a little introduction about the speaker, Dr. Indrapal Singh. PhD in Organic Chemistry from Punjab Agriculture University, Ludhiana, and a PhD in Natural Products Chemistry from Shizuoka University, Japan. To his name, there are numerous book publications like Stereochemistry, Analytic Profiles of Selected Medicine Plants. Number of patents has been granted to his name, so I request everyone to please put your hand together and welcome our speaker, Dr. Indrapal Singh.
Thank you very much for the nice introduction, and thanks to Professor Smith and the organizing committee of the conference for inviting me to deliver a talk here. So today I'll be talking on development of herbal formulations from sea buckthorn. So in the background you can see Naiper in the previous slides from Professor Sharma. Also you have seen perhaps the same photograph where we enter the Naiper, and towards the right side of this road we have the research blocks. So I'll give you a first brief introduction about the work that we are doing in our lab at Naiper. So we are broadly working in 3 different areas. One is synthesis of bioactive natural products. The other one is isolation of bioactive natural products. And the third area of research is standardization of herbal formulations. In the synthesis of bioactive natural products and their derivatives for different therapeutic areas that we are working on are leishmania, cancer, and what we do is we choose bioactive natural products that have been isolated from different plants for those activities, and then we try to derivatize these by introducing different functionalities in those compounds to evaluate their activities and to establish the structure-activity relationships for those compounds. Some of the compounds like berberine, forskolin, bicolin, caffeoyl anilides, quinolines, all these compounds have been evaluated. We have synthesized perhaps several hundred of these derivatives and evaluated these for anti-HIV activities. Euglobals, naphthoquinones, fluoroglucinol dimers, these have been evaluated for anti-leishmanial activities. And furanocoumarins as well as several other compounds that I have not shown here in the slide have been evaluated for anti-cancer activities. The other major area of research is isolation of bioactive natural products, where we choose plants based on their therapeutic uses or traditional uses and then try to isolate active principles from these plants. So we have worked extensively on eucalyptus species, the leaves which we received from our collaborators at Australian National University in Canberra, and also from plants that we have received or collected or procured from market in India. So one of the plants is eucalyptus, both Australian and Indian species, and we have isolated a number of fluoroglucinol compounds, some of which have been shown here. The other plant is potentilla, which is commonly known as Vajradanti. It is commonly available in northeast India. From this plant, we have isolated few new compounds, no unknown compounds. Then Rhodiola imbricata is another plant on which we have worked extensively and isolated several new compounds and evaluated these for different activities. So Rhodiola imbricata has been called, among many other plants, also called as Sanjeevani booty. Then we worked on microorganisms also, like endophytic fungus, Lasiodiploidea pseudotheobromii, and we isolated few new compounds. Then the other aspect of our research has been standardization. So we prepare extract from different plants, quantify different marker compounds in those extracts, and also quantify these in different marketed formulations in order to standardize those extracts. So for this, we use extensively the analytical techniques like HPLC, HPTLC, and quantitative NMR. So today I'll be talking mainly about sea buckthorn, which is a high altitude plant, grows at a height of around 15,000 feet in Leh Ladakh region, as well as Lahaul Spiti region, and also the higher reaches in Sikkim and Northeast India. So this is a very widely wild plant growing alongside the roads when we go from Leh to Nubra Valley or to other sides of Leh Ladakh. So this plant has been used. There are many marketed products from this plant. So today I'll be talking about the development of the extraction techniques for sea buckthorn fruit oil and its evaluation as for wound healing activity, as well as development of different formulations from this fruit oil. Then we have worked on preparation of the polyphenol enriched fraction from the hydroalcoholic extract and evaluated this for anti-inflammatory activity. So sea buckthorn is a deciduous dioecious plant which is about 3 to 4 meters in height, and it grows in different parts of the world. In India, I have already told you, but along with that, it grows extensively in China, Mongolia, Russia, Canada, and all over the world there have been several marketed products. There are 7 species of this plant, which is common known as Hippophae. The species available in India are Hippophae rhamnoides, Hippophae salicifolia, and Hippophae tibetiana. So we had a project from Department of Biotechnology for development of formulations from this plant Hippophae. So this plant shows very good, remarkable healing properties, and it has very good nutritive values. It is very rich in omega fatty acids, or it is claimed to be very rich in omega fatty acids. The fruit oil contains these fatty acids, and it has been used in as a radioprotective when there was a nuclear accident in Chernobyl in Russia. The seeds of sea buckthorn are rich sources of proteins, and also flavonoids and other compounds are present in the oil. So these are basically the different types of compounds or natural compounds which are present: primary metabolites and secondary metabolites, amino acids, organic acids, phytosterols, terpenoids, minerals, flavonoids, fatty acids, carotenoids, and multivitamins. That's why this has been a very good source of a wide range of biological activities. So the activities which have been demonstrated for this plant, for different parts of this plant, are antimicrobial activities, antioxidant, ulcer protective, anti-inflammatory, wound healing, radioprotective, platelet aggregation, cardioprotective, lipid loading. So here are some pictures which were taken by one of my students during his visit to Leh Ladakh. You can see on the left-hand side the growth of this plant as small trees. Middle picture is the seeds, and the right picture is the fruits, which are very sour in nature, contains high amount of citric acid in them. So these are the regions where this plant grows extensively in Jammu Kashmir, Leh Ladakh, Lahaul Spiti, and Sikkim. So the major fatty acids or omega acids which have been observed in the oils from this plant are palmitoleic acid, palmitic acid, oleic acid, linoleic acid, and linolenic acid. The percentage of this oil in sea buckthorn seeds is around 8 to 20%, and in the pulp or fruit it is around 20 to 20%. The composition of seed oil and fruit oil is very different, and that's why they have different biological activities. So besides this, this plant also has vitamins, functional elements, and phenolic acids. So the major types of compounds include flavonoids, lignans. The percentages are mentioned here: 32%, 14%, 21% oils, tannins. These are taken from literature, and there is a range of these values in different literatures available. Besides this, several phenolic acids like protocatechoic acid, salicylic acid, vanillic acid, caffeic acid, and many others are also present. So there have been total 99 flavonoids that have been isolated from hypophyraminoids so far. So here are the structures of some of these compounds which have been isolated bioactive compounds from this plant. So these structures are shown for flavonoids and their glycosides. Now, when we saw the literature, we found that the fruits of this plant have either been used for preparation of the oil or for the phenolic enriched extract. We could not find any literature where the same plant material has been used for both the preparation of the oil as well as the phenolic enriched extract. So we wanted to develop extraction techniques where we can use the same plant material for its optimal utilization in order to obtain both the hexane fraction, that is the oil, as well as the polyphenolic enriched fraction. So there were two routes: either we first extracted with hexane or we first extracted with ethanol. So we adopted both the routes. So first we prepared the fruit oil and then prepared the ethanolic extract, and in the alternate route we first prepared the ethanol extract and then partitioned it with hexane to prepare the oil. And then we compared the extractive yields of the fruit oil as well the ethanol hydroalcoholic extract prepared, and we could find that the extractive yields of both the oil as well as the ethanolic extracts were very similar. So therefore we adopted route 2, where we can first extract the same fruits with hydroalcoholic extract so that it will not have any traces of hexane in it, and then it is followed by extraction with hexane, so to have the optimal utilization of the fruit materials. So after preparing these oil and extracts by two alternate routes, we did their TLC profiling, as you can see in figure on the left hand side. So is the TLC of pulp oil and the fruit oil, H1 and H2. So either we separate the seeds from the fruit, separate the pulp, or we extract the oil. The TLC was very similar, so it did not matter much. So then we compared the extract and the ethanol water extract prepared by the two routes E1 and E2 and H1 and H2, and we could see that the TLC showed very identical profiles for both these extracts. So that indicated that both the extracts prepared by both the routes, they had very similar metabolite profiling that enabled us to select route 2 for the further studies. Then the further studies were on standardization of these oil and the extracts and their formulation development. So first I will talk about the standardization of oil and its formulation development and evaluation of the wound healing activity. So it is reported in literature that sea buckthorn oil is rich in omega-7 fatty acids, which are rare monounsaturated fatty acids offering unique health benefits. And seed oil is rich in linolenic acid, that is omega-3 polyunsaturated fatty acid. So all the marketed product, fruit oils or oils which are available in the market, they claim on their labels that they are rich in omega fatty acids. But when we started working on this fruit oil for isolation of these compounds, instead we isolated large amount of a compound that was about 70% of the oil, which showed the clear signals from triacyl glycerols. So that suggested to us that the oil was rich in triacyl glycerols, not the free omega fatty acids. So in the NMR, we could see signals at chemical shift of 4.12 to 4.3 and 5.23 to 5.28, which can be attributed to the alpha and beta protons of the triacyl glycerol. So then we try to quantify the amount of triacyl glycerol by using quantitative NMR, using the equation which is shown here in the slide, which takes into account the integral values of the signals, the molecular weights, the number of protons that we are considering in the structure for quantitative purpose. So simply putting these values in the equation above, we could find that 11.4 milligrams of the oil contained about 9.1 milligram of triacyl glycerol. So the omega acids are not present in free form, they are present in the triacyl glycerol form, and when this is ingested, then probably they are hydrolyzed and we get the free omega fatty acids. So the results showed that the tag percentage in fruit oil was about 80%. So then we wanted to identify and quantify the side chains of the triacyl glycerol, and it is reported in literature that for these fatty acids, we can estimate the amount of those linolate, linoleate, palmitoleate or saturated functional groups by using the equations which are shown in this table. And we took this table from literature and found that this oil which we prepared from fruit, it contained linolenate about 4.7%, linoleate about 5%. The major amount was palmitoleate and oleate, that was around 60%, and saturated acyl chains constituted about 32%. Then we looked into the literature and we found that the fruit oil contains large amount of saturated fatty acids and palmitoleic acid, say 28, 29% and about 50%, whereas the seed oil is rich in linoleic acid and linolenic acid. So then we purchased commercial sea buckthorn fruit oils from Amazon.com from 6 different vendors. Except one of these, no vendor had mentioned that whether it was a fruit oil or seed oil. As we can see from literature, their composition is entirely different. So we used quantitative NMR for the quantification of these triacyl glycerols in these commercial oils, and we found that all these commercial oils contained large amount of triacyl glycerol and not the omega fatty acid, free fatty acid. And one of these, which was imported, that was at serial number 2, it had a different composition, while the Indian oils had a different composition. So quantitative NMR was used to quantitate this omega-3 and omega-7 as well as saturated acyl chains. And we could see that the fruit oil which we had prepared, that contained about 65% palmitoleate, and the imported sea buckthorn oil contained about 64. All other from Indian vendors, they contained about 22 to 37% palmitoleate, but they contained larger amounts of linolenate and linoleate. So this suggested to us that all these oils, they were either mixture of fruit oil and seed oil, or they were seed oil, but it was not mentioned on their labels. So we also did this analysis by GCMS, and we were able to identify the omega fatty acids by using GCMS. So most of the quantification studies on these oils are done by GCMS, and during this GCMS analysis, these triacylglycerols break down into the omega acids, and that is what had been reported prior to our work. So these are the results from GCMS analysis indicating the presence of those omega fatty acids. Then we evaluated the wound healing activity of sea buckthorn oil, and as we can see, A is the negative control, B is the positive control, that was fremicetin, sulfate, and C is the fruit oil. So we can see a very good wound healing activity of hypophyraminoids fruit oil at different concentrations. We applied 100 microliter, 200 microliter, and 400 microliter, and this we could see good wound contraction by application of the seed oil, and it was comparable with the standard fremicetin sulfate. This biological evaluation studies were done in the lab of our collaborator, Professor K.B. Tikku. So these are the results showing the contraction in the wound area on day 4, day 8, and day 10 at different amounts applied to the wounds which were created on the animals. So then after evaluating the biological wound healing activity, we wanted to develop the topical formulations, and we developed cream and gel formulation from the fruit oil, and for that we first prepared the nano emulsion from the fruit oil using 10% of the fruit oil, 1 to 5% of surfactant and co-surfactant, and then making up the volume with water using a probe sonication for 4 minutes. And we characterized this nano emulsion by evaluating the droplet size and PDI values. I'm not showing those details here. And after preparing the nano emulsion, we prepared the nano emulsion loaded cream using the ingredients water, glycerol, glyceryl, monostearate, caproyl, cocoyl caproate, tween, and the nano emulsion of the fruit oil. The percentage is shown here in the table, and the procedure as shown here. So both were heated to about 45 degrees and then cooled, mixed, and then we prepared the nano emulsion. So same way we also prepared the nano emulsion loaded gel from the fruit oil using carbopol, sodium hydroxide, and hydrolyte in the amounts which are shown in the table. And then both the formulations were again evaluated for wound healing activity. So here we are showing the groups that were, animals were divided into these 6 groups. One was disease control, negative control, positive control, that was fremicetin sulfate, then hypophyraminoids, nano emulsion gel, and nano emulsion cream. So wounds were created on dorsal surface, and then the oil and cream and gel were applied on these wounds. And as we can see here, after 10 days, there is almost complete healing of the wounds by application of nano emulsion cream, nano emulsion gel, as well as nano emulsion, and it was better compared with the positive control. So we also did the ex vivo skin penetration studies. As we can see here, the cream and gel were able to penetrate deeper into the skin, resulting in a better wound healing activity. So we can see here that green fluorescence due to the coumarin C6 which was added. So that green fluorescence is seen in D and C, that is better, showing a deeper penetration of cream and gel into the skin. So then the cream and gel were evaluated for acute dermal toxicity studies, and we could see that they did not cause any adverse reaction and they were safe in category 5 of the OECD guidelines. Then the second part of the presentation is the evaluation of 70% ethanolic extract, its formulation development, and its evaluation for anti-inflammatory activity. So these are the compounds that were isolated after doing extensive chromatographies, including vacuum liquid chromatography, flash chromatography, and HPLCs. These compounds were characterized by NMR data, mass spectrometry, and IR data. I'm not showing that characterization data, but these compounds were isolated from sea buckthorn fruit ethanolic extract. And then we worked on enrichment of the ethanolic extract. We prepared the polyphenol enriched fraction by doing chromatography over XADHP7 resin. So first it was washed with water to remove the sugars and then eluted with methanol to make the polyphenol enriched fraction. So qualitative analysis of this enriched fraction was done by LCMS and HPLC, and we could identify several compounds which included citric acid, quinic acid, and isoramnitin is the major compound, and isoramnitin glycosides as well as quercetin and camphorol glycosides could be identified in the polyphenolic enriched fraction, and as per the phytopharmaceutical guidelines, we quantified these 4 markers in the extract. These are the characterization data using LC-MS of those 4 glycosides. Isoramnitin glucoside, isoramnitin rutinoside, and quercetin rutinoside were characterized. We can see the loss of glucose, rhamnose, and pigments were identified which helped in characterization of these compounds. So then this ethanolic, polyphenolic enriched extraction fraction was evaluated for total phenolic content as well as total flavonoid content, and we could see an increase in total phenolic content as well as total flavonoid content after enrichment. So these were done by the usual established methods for determination of phenolic and flavonoid contents. HPLC quantification for these 4 compounds was done, and we could see the enrichment of quercetin, almost 10-fold enrichment in amount of isoramnitin, and large amount increase in amount of isoramnitin thioglucoside when we compared ethanolic extract as well as the polyphenol enriched fraction. So this polyphenol enriched fraction was then converted into its phospholipid complex by the established methods using phospholipid 90H, and this phospholipid complex was characterized by NMR and IR studies, and we could see the upfield shift in NCH3 protons after preparation of the phospholipid complex, which indicated that the phospholipid complex has been formed from these flavonoids or this polyphenolic enriched fraction. So here I'm showing the possible interactions that may have happened in the formation of this phospholipid complex, and it was established through the shift in the chemical NMR chemical shifts as well as the shift in the IR absorption bands of these compounds. Then we subjected this phospholipid complex for in vivo anti-inflammatory activity using 50 milligram per kilogram and 100 milligram per kilogram body weight. And we can see that this polyphenol enriched fraction phospholipid complex showed very good inhibition and very high anti-inflammatory activity in the paw edema assay. As we can see here, up to 70% inhibition after 5 hours was seen in the rat paw edema. Then after evaluation of this activity, we subjected this phospholipid complex to stability studies, which included long-term stability. We kept it in refrigerator and also real-time room temperature studies as well as accelerated studies, under accelerated conditions as are the guidelines given in the ICH stability testing guidelines. And we could see that the formulations was fairly stable. There was slight decrease in the glycoside, which is generally hydrolyzed into the A-glycone as well as the sugar moieties. So that was the study up to stability that we have done, and after that we did the bioavailability studies, which is not, I'm not presenting here. So in conclusion, we can say that we have established methods for standardization of fruit oil as well as the ethanolic extract and the polyphenol enriched fraction. So we could say that the fruit oil contains triacylglycerols and not the omega acids as is claimed on the labels of the most of the marketed products. Then we prepared the formulations of oil and ethanolic extract and evaluated these for different activities. So this was, a study was funded by, it was a multi-institutional project. The plant material was supplied to us by IHBT Palampur, and we had co-investigators from NIPER, Professor Tikku, Professor Jachak, Professor Jaina for doing the biological studies, and most of the chemistry studies were done in my lab, and couple of PhD students and one postdoc worked on this work. And here you can see Dr. Datta, who is present in the audience today. He did most of this work on chemistry and biological evaluations of this plant. With this, I thank you very much for your kind attention, and I'll be happy to answer any questions that you have. Thank you very much.
Thank you, Professor IP Singh. That was an exhaustive talk. You've covered everything. I think the students would be able to understand he has covered the complete spectrum of drug discovery and development from plants, right from collection to processing to getting a formulation and studying their efficacy and safety. It was such an exhaustive talk. Thank you so much, Dr. Singh.
Thank you, Dr. Saab, for apprising all of us regarding the development of formulation of sea buckthorn, wonder berry और लद्दाख बेरी भी कहते हैं इसको, या ले गोल्ड के नाम से भी शायद जाना जाता है. And the session is open for discussion. So anybody if have any question can ask.
Among the audience, if they are having any sort of query or question, please pose.
या तो सब कुछ क्लियर है या बिल्कुल कुछ नहीं समझ में नहीं आया.
Can I ask you a question?
Yeah, sure.
जब आपने, when you prepared the extracts, petrol, were they successive?
Was
the
that?
ethanol, the alcoholic extract and all, were they successive from the same
No.
or
So
separate?
we prepared the ethanolic extract, only 70% ethanolic extract from the fruits using Soxlet, and then the remaining material, we used the extract as such, subjected it to chromatography to prepare the polyphenol enriched fraction. The plant material which was recovered, the fruits, after the extraction, they were then subjected to preparation of the oil, so that hexane does not come into the ethanolic fraction.
Right, sir. And there was another thing, you mentioned that you have worked with fruit and the seeds, the pulp.
So
Did the fruit include the pulp and seeds?
initial study was done with fruit as such without removing the seeds. And then the study was done after removing the pulp, and we compared both. The profiles were very similar because the amount of seeds is very less in the fruit. So then we worked, the rest of the study was done with fruit intact, with seed intact in that. Yeah.
Thank you. Anybody else would like to ask anything?
Sir, in the slide number 25, sir, you have mentioned about the peaks of camphorol and glucoside that was in the combination, and it was the second largest in the peak as compared if you see in the diagram, sir.
So that was this slide you are saying?
Yes, sir.
Yeah.
Camphorol, glucoside, rhamnoside combination, sir. That one, sir.
Yes.
Sir, just a simple question. It was a fruit extract based, ethanolic extract based experimental design for the purpose. What if that same camphorol, as if it is a portion of my research work also, is it is actually in a silico based way in a combination with a molecular docking. The percentage was around 76.28% on the protein CY3C. Sir, what would be the comparison if we do that ethanolic extract as compared with the in silico or molecular docking procedure? What will be the percentage yield if you compare both the phenomena, sir, for a particular target?
No, so actually I could not exactly get you. So in silico study, what you have done is 76%. What was that 76%?
Sir, that was the docking potential, or you can say receptor drug
docking potential with what?
Yes, of all those drugs, particularly non-steroidal anti-inflammatory drugs,
Okay.
working on the action of AKI, acute kidney injury.
Okay.
Sir, target was kidney injury expression type 1 biomarker. Sir, 6Y3C.
Okay.
6Y3C.
So, say, in silico, when you are doing the studies, we do not do in silico much, but when you are doing in silico studies, you are doing the docking against one target or maybe two targets. But in actual practice, you have all the targets available where these molecules may be binding to different extents. And the overall result may not be the same as you get in your docking results.
Yes.
So that can give you some indication that, okay, well, it may bind with this protein or this protein, but we do not know all the proteins with which those may be binding in actual. Okay, so I think the docking results comparison with the actual results may or may not be comparable.
Okay, sir. Thank you, sir.
Yeah.
That's all.
Anyone else from the audience side with a question?
So I think there is no question now. So the session, we can conclude the session.
Yes, sir.
Very wonderful talk, Dr. But I just wanted to clarify one thing, that when you extract plants, I have heard, I may be wrong, that the actual content, actual phytoconstituents might change, the structure might change. This is called extraction artifacts. So how will you confirm that the content or the phytoconstituent you have extracted is the real one or the changed one?
Yes. So very good question. So there are several ways to determine whether it is artifact or not.
Yes.
So one is that we extract it with very mild conditions and then identify that compound in the extract before doing any chromatography or processing it further.
Okay.
So if it is present when you extract it at room temperature or at low temperature, then we generally say that it may not be an artifact. Second is the thinking, biogenetic thinking. Sometimes the rearranged products that are formed, we may not explain these by any biosynthetic pathways. If we cannot explain, then perhaps they are artifacts. Yeah.
Thank you.
Thank you.
Thank you so much, Dr. Inderpal Singh, for sharing your thoughts and insights and as well answering the questions and queries of audience with such graceful manner. I take this opportunity and request the chair session members, Dr. Richa Shri and Dr. Nitin Bansal, to please present a token of appreciation to our worthy speaker, Dr. Inderpal Singh. Please join us on the stage. Thank you so much. I cordially request Dr. Manisha Bhatia to present a memento to Dr. Richa Shri. Please join us on the stage. Thank you so much. I invite Dr. Sukhbir to join us on stage and present an appreciation token to Dr. Nitin Bansal. Thank you so much, dignitaries. Ladies and gentlemen, we'll be proceeding towards the third lecture of the day, which will be delivered by the respected speaker, Dr. R.K. Goyal, DSPDR, Punjab University, Patiala. I may request Dr. Inderjeet Verma to come forward and present a welcome sapling to Dr. R.K. Goyal. Both the esteemed dignitaries, please join us on the stage. Ladies and gentlemen, we'll be waiting for our chair session members to join us shortly. Ladies and gentlemen, it gives me an immense pleasure to inform you all that this lecture will be chair session by Dr. Subhit Jain and Dr. Suresh Thakur. For the same, I request Dr. Jasmine to welcome one of our chair session members, Dr. Subhit Jain, on the stage. I request Dr. Jasmine to please welcome one of our chair session members, Dr. Subhit Jain, on the stage. Thank you, sir. I humbly request Dr. Anuj Malik to present a sapling to Dr. Suresh Thakur. Please join us on the stage, sir. Thank you so much. For the information of the audience, our worthy speaker, Professor Dr. R.K. Goyal from Punjab University, Patiala, has the areas of specialization in neuropsychopharmacology, phytopharmacology, pharmacogenomics, and computer-aided new drug discovery using PASS. He has done the bachelor's in pharmacy from Amravati University in pharmaceutical science, master's from Punjab University, Chandigarh in pharmacology, and doctorate from Punjab University, Chandigarh in pharmacology as well. He's been the member and is still has the membership of professional bodies and organizations. President at APTI.
That they will be having less effort for the approval as new chemical entity as compared to new chemical entity, and there are certain conditions for what we say approval of a plant extract as a phytopharmaceutical, which will be useful for treatment of diseases. But if we talk about advancement of drug discovery from herbal drugs, so we have nowadays talking about in silico techniques which are useful in number of ways, and these are also applied to the herbal drug discovery. If we go back to the history, you'll find that Indian medicinal plants, they are the backbone of the drug therapy. Before the synthetic era, 80% of the medicines were obtained from botanicals, maybe barks, roots, fruits of different plants. And India is richest. If we talk about the flora of India is richest, we are having about 120 families of plants which are comprising of 130,000 species. And in Ayurvedic texts, about 2,400 species of the plants with their medicinal uses has been mentioned. These are described there. So 10,000 herbs are used worldwide for medicinal purposes regularly. And I told you as earlier that 80% of the drugs were from the herbal resources before the synthetic era. So, but there was sudden decline in the interest of medicinal plants during the synthetic era. Why? Because there was advancement of synthetic organic chemistry and the small molecules were taking place instead of the extracts, and they were found to be more effective, they were found to be very potent, and because of that and certainty of their effect, you can say, transformation from the Ayurvedic to the modern medicine was going on, and these advances lead to development of good medicinal chemistry and process chemistry, and these aspects lead to the development of what we are taking nowadays as a modern medicine, the single molecules which are effective for with a known mechanism of action on a particular receptor that is there. So pharmaceutical industry was established to flourish this concept rather than taking forward the plant extracts or the plant-based medicines. The processing of medicinal plants was cumbersome. That was one of the reasons that people were not talking about taking plant-based medicine forward. sourcing authenticated plant materials, inadequate documentation and loss of original plant collections was there. And if we consider that as per the Vedas, we are having very good information about the Ayurvedic, you can say, use of different plants, but that information is missing. We are not having comprehensive information as of date, so some part of it is missing. So lack of measurement of biological activities. So what modern medicine says that we should have a standardized activity of a plant extract. It should not be like that. If we are taking one plant from one region and extracting that, and other plant we are extracting from the other region, the activity can be different because of, we know, the different variations that lead to what we say, the changes in the phytochemical or phytocomposition of those extracts. So that can affect the issues. Then purification and identification of active constituents again was a cumbersome process at that time, but now technology has taken a lead and we can think of this. And supply to meet the market demand, that was a bigger issue. We can have, you can say, in the local area you can have some plants and use that for medicinal purpose, but you cannot make it global. That was the reason. So that globalization of plant-based medicine was a difficulty because of that gap of supply and demand. But somehow there was a renaissance in the medicinal plants. People have, you can say, catch up with the lost interest in the medicinal plants. So because in the synthetic medicinal chemistry, we were not able to find new structures. So nature always gave us a template, a new template structure which is useful for the medicinal purpose, which was not possible through the synthetic processes because we have a finite thinking. We do not have a infinite thinking. The molecules which we are talking about nowadays, and if we are trying to modulate or you can say modify those molecules, we are not going to get very, very new molecules because of that. So difference of high cost of modern allopathic medicines, advancement in analytical and biotechnological and pharmacological techniques in the herbal drug discovery, these things led to renaissance of our interest in the herbal medicines back on the plants. So if you see, this data is a little older data, but if you find that natural drugs, and if you see, are 67.6%. And this is how the things changed. There was little low interest, if you see between 92 and 2000, and thereafter, the percentage of drugs, new drugs, was very less. So we have to look back on the, what we say, the plants as a source of new drugs. So what are different approaches for drug discovery from medicinal plants? There's random approach. And if you see, most of the colleges, when we go for thesis examination, they say we have extracted one plant and we have tested it for anti-inflammatory activity or blah, blah. There are a number of activities which they are going to check for that. So screening of randomly selected plants for selected bioassays is common thing. And this is called random approach. And disadvantage is that we need a library of extracts. So we should know what are different type of extract, what can be the constituents. If we do not know, then we cannot have the rational approach on that. Then ethnopharmacology approach is there. Most people say that this plant is used in the ancient books for this particular purpose, so that's why we are trying to use it in the modern medicine for this purpose, and we are trying to use animal models to validate that use. Then reverse pharmacology. Reverse pharmacology is we take in two ways. In this context, what we talk about, whatever we know about the plants, we are trying to have a reverse engineering to know that which phytoconstituent is useful for that activity and what is the mechanism of action. That's reverse pharmacology of the plants. The traditional system of medicine approach is there. Zoopharmacognosy approach. This is a very, you can say, old approach whereby it was monitored that which type of plant animals are taking when they are having certain type of problems. So based on that, they are saying that this particular plant may be useful to take care of that disease. So phylogenetic clustering is another way to find the, for example, we are having a family of a plant. That family may be having similar phytoconstituents. Based on that, we can say that this can be useful for similar type of pharmacological activity. But all these approaches were limited to explore the known effect of the drugs, or known effect of the plants. They were not going beyond it. So when we say that if you see the phytochemical space, that has not been explored fully till date. Only about 2,000 species we have seen, which is mentioned in Ayurvedic text, from like 1,30,000 species. So if we see this way, that means we are having a lot of potential to get more uses of the plants which are available with us. So this can be carried out if we look beyond the ethnopharmacology for the source of new drugs from the medicinal plants. So that was the source for this particular presentation. I'll skip some of the things because they are not directly related to you. So this is another approach where bioinformatics is playing a role nowadays, where you can find out a particular phytoconstituent and then you can find the targets for that, and you can match the targets and the phytoconstituents to find new uses of these molecules. So Ralinger et al., if you see, they say that if we have to use in silico techniques, so in silico techniques have three components. One component is we should have a database. Okay, we should have a database, and if we have a database, then we should have a logarithm to get the prediction of new plants based on that database training data set. So we are having molecular talking as approach. Then we have pharmacophores, quantitative and qualitative structure activity relationships, and machine learning methods. So all these are taking a seat for new drug discovery based on plants. So as far as databases are concerned, there are a number of databases. If you can see on the screen, we have a lot of databases. Now these databases are, for example, Ayurvedic medicinal plants of Sri Lanka, database of ethnomedicine plants. So I will not read all these things. You can see the number of databases which are available. And Impetat, if you see, Impetat 2 is the recent one, which is from Institution of Mathematics Sciences. Okay, so this particular, I'm a fan of this particular database because it is having a lot of information about medicinal plants, the structure of phytoconstituents they have, and the activities reported till date, and Ayurvedic uses, and so other things. So if we talk about docking and other things, so how many of us are having the facility for docking in your institutes? Very few institutes are having this problem because of the high cost which is involved in procuring the softwares and other things. So in this context, what we did, we collaborated with the Russian team, and they were having a software PASS, which will depict on, and that is having ability to predict based on 2D structure of the molecule, and it is giving number of other activities, not only single activity, because in the docking we are supposed to have only single activity to which the docking is with the target which is being docked. So based on this, we have moved on to 2D approach. So this is PASS, which we have used in different synthetic molecules to predict their pharmacological activity, and that was validated in the lab for their activity. So 2D based in silico tools for prediction of biological activity, we are using two tools. One is PASS, other is Pharma Expert. PASS is giving us the possible effects, and then those effects by which mechanism they can be possibly showing their effect or exerting their effect, that is based on the prediction in the Pharma Expert. So these are the two different softwares which are used for this purpose. So a little bit about PASS. PASS is the prediction of activity spectra of substance. So its training set is always evolving and increasing, and because of that you will have better efficacy with time. So as of now, this 95% of accuracy of this particular prediction made by this, and the score, if you see, if P is less than 0.7, then there is very high probability that the molecule will be having the predicted activity. At the same time, the novelty of the molecule is less, and if it is between 0.5 to 0.7, then the chances of activity are fair enough, and if it is less than 0.5, then we say the activity, chances of finding the activity experimentally are very low. But if we find that activity experimentally, in that case, the novelty of that molecule will be very high. So this is how we work on this. So if this, in this slide, we are trying to depict that if PAS and traditional Indian medicine plants, they are collaborating, and then we can definitely have some novel pharmacological activities for the phytoconstituents vis-a-vis of plants, and those will be useful. Those can be verified in the lab, and we can then have repositioning of the plants. In this regard, we had also prepared a database of 50 medicinal plants, which is available at the, I'll just show you the slide, waytodrug.com with plants. So this website is hosting this particular project, and this is being, there's a database of 50 medicinal plants which we made as a trial, and it is available there. So it was retrained for Ayurvedic, you say, medicinal plant-based phytoconstituents, and then again the accuracy was very good. So coming to validation, for example, if we talk about, so this is the prediction coefficient of PAS, which we have retrained, and you will find the last one. The one means we are trying to compare what are the known activities which are being predicted by the software, and if it is predicting almost all the known activities till date, that means the prediction coefficient will be 1. But in the last column, if you see, some of the plants were predicted with the prediction coefficient of 1, and most of them were having more than 0.6. So that means the predictivity of the new PAS software is better for the medicinal plants based on phytoconstituents. So predictive biological activity spectra of 50 plants, if you see, this is there. So are you able to see, differentiate the color? There's green, light gray, and the light gray and dark gray. In the base there is dark gray color. On the top it is light gray color. The base dark gray color shows the known activities, and the upper one, the light gray, that is showing the unknown activities. You'll see that there are many plants which are showing a large number of activities which are not known till date, but they are predicted by this. So this was the aim of this particular project. So we are trying to find out, explore the unknown activities of the medicinal plants which can be validated practically. So this was published in this particular journal, this work which we have done for database creation. So then another thing which we talk about is a case study. I was thinking of we should talk about a case study. Here we are talking about pharmacological validation of in silico guided novel nootropic potential of Acanthus aspera. So this plant is having traditional uses in asthma, cough, diuretic, purgative, and laxative. It is also used in piles, boils, and eruption of skin, liver complaints, rheumatism, scabies, other skin diseases, and anti-allergic to wasp stings. So these are the traditional uses of this plant. But when we got it predicted with the help of PAS using different phytoconstituents, we find that many of these traditional uses have been validated pharmacologically. In addition to that, hypolipidemic effect can also be seen, but no major CNS related activity was mentioned in the literature. So based on that, we thought that can this plant be explored for some neuropharmacological activities or not. So this is the number of phytoconstituents which were used for predicting whether it is having some nootropic effect or not. You will find that the three major, you can say, effects where number of constituents was shown to have this effect of improvement of memory, either they are useful for dementia treatment, vascular dementia, or they are termed as nootropic in the PAS predictions. So this is the table which you can see for the PAS predictions. And once we came to know that this is possible, that we can have nootropic activity, because the score was very high, it was more than 0.7. So then we tried to validate this particular plant extract, which was what we say, standardized with the phytoconstituent which had shown their you can say effect as nootropic. So this is the scheme of pharmacological experiments whereby we are talking about different materials used, and then animals and plant extract. So these are the doses of Acanthus aspera. Paracetam was used as a standard drug, and betaine was used as a standard herbal drug drug, whose content was monitored in the extract, so that we can see what is the change in the effect of the extract with the dose. So standardization of Acanthasis aspera root extract was with respect to betaine. It was performed. So this is the experimental protocol whereby the treatment days, if you see, we started locomotor count in -3, 3 days before the training, and then retrieval testing of what we say locomotor activity and passive awareness test was carried out for the memory evaluation. It was step through passive awareness, light dark chamber was used for that. Then we had radial arm maze training, and it was used for reference memory errors and working memory errors. So 6 groups of animals were used, and this is how the grouping was carried out. And this is the result for standardization of the extract with the betaine. And based on that standardization, we have used the doses of extract also. Dosing of extract was based on the betaine content. And then effect of different pharmacological intervention, if you see on the spontaneous locomotor activity, there was no significant effect of betaine as well as Acanthasis aspera extract. So, but when we talk about the working memory errors and the radial reference memory errors in the radial arm maze, there was a significant effect of these extracts, both on working memory errors as well as reference memory errors. And the retrieval latency, if you see in case of radial arm maze, that was also highly decreased, latency was decreased, so that shows that memory has been enhanced. It was almost comparable to paracetam in that case. So different, you can say, we have also tested novel object recognition in that. Novel object recognition test was also having better recognition index with the treatment of Acanthasis aspera as compared to the control. Then we have passive avoidance test. In this case also, if you see, the step through latency was very low on training day 0. On retrieval time was this day 1. It was enhanced, and in the 7th day it was again enhanced. So the latency was enhanced. That means the shock which we were providing in the dark chamber, that was avoided by the animals. So that means they were remembering that if we'll be going from light to dark chamber, we'll get a shock. So that's why the latency time is increased. That increase means they are remembering. So memory is improved that way. So this is again, we have made some neurochemical estimations in the hippocampal and cortical area, where they have shown that glutamate level have been little bit increased, or significantly increased as compared to the saline group, and it is dose dependent as far as Acanthasis aspera is concerned. So this is again for acetylcholinesterase activity we have tested, because that is also having a role in the memory enhancement. So it was also decreased by the Acanthasis aspera extract, but it was not dose dependent, but it was decreased, but it was significantly decreased as compared to betaine itself. But that means there are some other constituents also which are bringing down the acetylcholinesterase activity, and they will be useful for making the acetylcholine availability in the brain better. So with these results, we can conclude that the in silico predictions made by past and pharma expert has resulted in repositioning of Acanthasis aspera for the dementia or for improving the memory, which was not earlier in the literature. So we can repurpose the animal, plants based on this technique. That is one way. And then we can also determine number of other things. We can find out what is the most active phytoconstituent in extract which is showing the effect. That is one thing we can go. Secondly, we can understand what is the mechanism based on which type of pathway it is going to interfere. That can also be, because this is not clear in most of the Ayurvedic medicines, that how they are acting, what is their mechanism as per the modern medicine. So this can also be taken care of with this approach. So these are some of, this paper was published again in Ethnopharmacology and for anticonvulsant activity, and then we have number of other publications based on this approach. So I acknowledge the grants given by DSTRFBR for this collaborative project, which was Indo-Russian project. Thank you very much for your attention.
So thank you, Dr. R.K. Goyal, to give such a nice and informative talk. Sir has explained that how these in silico techniques are going to be useful for validations of these natural plants also. And this is something very new, because most of these students are working for either for formulation development or either for means evaluating the different pharmacological activities using the medicinal plant. So this is going to be, sir, very beneficial for whole audience. And in addition to this, because India is known for this natural means isolates and all natural drugs, so this is also very good for future validations of our Indian plants to be used as a drug also.
Yes.
Another things that I want that here, that nowadays in vivo studies, most of the regulatory and the CPCSE is saying that you have to reduce the use of animals.
Yes.
And probably once we will run the things with your this model, probably we need to use the very minimum number of the animals, and probably the data of the animal experimentation is also going to be acceptable, because the latest report is saying that 50% data is not reproducible. It is not the fault of all of us, it is the natural variability is there. So probably this use of both pass as well as design software and how you explained us to use it, it's going to be highly beneficial for whole scientific community. Now this session is open for discussion, and followed by I will give this mic to my fellow colleague Dr. Suresh, because he is expert in this field. He will further tell you.
So first of all, session is open for any query from students and colleagues sitting here.
Any questions?
Any question from audience? Sir, I have one query.
Yeah, please.
As a plant is complex mixture of constituents.
Yes.
So this PASS software can help in locating the bioactive constituent in a crude extract, because as a plant is a complex mixture of constituents, and different constituents have different multifaceted activities, different activities. So which constituent is located, considered to be a marker compound for standardizing a plant material? Can
Yes.
PASS help in?
Yes, that is what we have done. Uracilic acid we have used from what we say, tulsi, and it is useful as hepatoprotective. The hepatoprotective activity was attributed to uracilic acid, and we have used uracilic acid as such to see what is the potency of that hepatoprotective effect in comparison to the extract. And we found that it was corresponding to the uracilic acid content. So we tried to see that thing also. So definitely it can give you the marker compound on which you can play for long. Like as in phytopharmaceuticals, we have a condition that we should know at least 4 phytoconstituents which should be standardized in that extract for being a form of phytopharmaceutical. So if those 4 phytoconstituents are responsible for bioactivity, it is much better, rather than having a you can say house type of housekeeping alert that this content is there. If it is related to the activity, that is more better.
Preferably these should be bioactive compounds.
They are bioactive, but they should not, they are all not related to, till date they are saying 2 should be related to bioactivity, 2 can be others. So we can, with this we can have 4. Okay. All the 4 which can be related to, or we can at least give the significance of monitoring at least that phytoconstituent in the phytopharmaceutical, that this is most important because it is monitoring the bioactivity. So it is responsible for bioactivity.
Yes. One submission is mine
Yes.
in this. Can we develop this software in such a way it will work as artificial intelligence to study mechanistic in way of synergism, antagonism associated between different constituents.
The limitation of this software is that it shows that it can have this effect, okay, or it can have affinity for this particular thing. Plus minus is not very good as of now. Plus minus is not very good, but when it is shown that it is anticonvulsant or it is useful for dementia, okay, or it is having nootropic effect, then it is definitely having positive effect. And second part of that software is Pharma Expert, which talks about what is the tentative mechanism that it is exerting the effect. We have two stages of validation, then we can go for that. And we can even validate based on using some agonist antagonist whether this mechanism is working for this particular plant or not. So that is also important. So then once we have, you can say the structure of that phytoconstituents, we can modify it and semisynthetic, you can say chemistry, and we can have better molecules for the activity. That is it.
Thank you, sir. Thank you.
Thank you so much, sir, for delivering such a knowledge. And thank you, our chairpersons, for your insights and thoughts. Now I'd like to call our chair session members, Dr. Subhi Jain and Dr. Suresh Thakur, to present a token of appreciation to our speaker, Dr. R.K. Goyal. I invite Dr. Sarita Sharma on the stage to please felicitate our chairperson member, Dr. Subhi Jain. I invite Dr. Sheikh Karimullah to please come up on the stage and felicitate our chairperson member, Dr. Suresh Thakur. Thank you so much to all the dignitaries. Now moving towards the next lecture of this session, which will be delivered by Dr. Neeraj Kumar Singh, Deputy Librarian, Punjab University. The chair session members for this lecture would be same, that is Dr. Subhi Jain and Dr. Suresh Thakur. I cordially invite Dr. Omveer on the stage to please present a sapling to our speaker, Dr. Neeraj Kumar. Ladies and gentlemen, our next speaker, Dr. Neeraj Kumar, has completed an international course on ICT from the University of East London, recognized by British Computer Society London, UK in 2012. He has been awarded as the University Librarian of the Year Award in 2021 by MPLA. He has designed and developed the chemistry portal, for which he was decorated with the prestigious Best Service Award 2009, presented by Vice Chancellor, Punjab University. And again in 2012, a portal on handling of hazardous material. He has numerous research papers to his name and the member of various organizations and committees. I request everyone to please put a welcome to our speaker, Dr. Neeraj Kumar Singh.
Very good afternoon to all of you. And before I will start my lecture, I would like to thank the organizer, specifically Professor Sumit and Dr. Seema Bansal, for inviting me and giving me this opportunity to share some of my knowledge on this topic, which I think is very, very important for the young researcher. I can see many faces, specifically from pharmacy students and the PhD scholar, and this lecture is going to be very, very useful for you. So I will request you to please open your notepad and pen, because whatever tool I am going to discuss with you, it's very relevant and very useful for all of you. You know, one thing very, very sure for research is the research needs to be disseminated. That is why researchers all over the world, they are publishing their research. And where they are publishing their research, they are publishing their research in various journals. So these journals are basically a platform which provides you a platform where you can disseminate your research finding. And one thing is also very sure in academics, that is called publish or perish. So if you are continuously publishing good research, then only you can survive in this academic world. You need good research paper for your recruitment, for your promotion, for your awards, for rewards, and even institutions, wherever they are applying for various ranking agencies. One of the important parameters is your research publications and citations. So without research, it is very, very impossible for you to build your career in this academic environment. So in this talk, you know, we are all witnessing the power of AI. So coming to the presentation, here you can see that there are many tools available for the researcher which are very, very helpful for you in each and every stage of your research. We are talking about AI, and you name any sector or any industry. I think all the industry and the sectors, they are using the benefit of these AI tools. So how the scientific community can be not touched on this particular area? So there are very sophisticated tools which are designed with the help of machine learning, deep learning, which we called as AI tool, which is going to help the researcher not only completing your research journey in a stipulated time, but with the help of these tools, you can also do the qualitative work. So in this talk, I am going to give you a brief overview about these tools. Like there are many tools which can help you in terms of searching the literature. You know, the research starts with literature review. And we all know that if you are not accessing the quality resources, then it will be very difficult for you to produce the quality work. So generally, library resources are considered as a quality research material for all the research scholars. But there are many students who are studying in different universities where you will not find access of some quality databases like Scopus, like Web of Science. These are the tools which can help you in terms of finding the relevant literature for your topic. But with this emergence of AI, there are many free tools, open access tools available, which can help you in terms of doing literature review with a very short period of time. So what are these tools available and how you can access these tools? That I'm going to discuss with you. Then there are various paraphrasing tools are there. You know, you can't write a complete PhD thesis or research paper of 10 or 8 pages without referring any sources. Because the topic of this sponsored conference is, one of the topic is research ethics. So we have to be very, very careful. The concept of the issue of research integrity, publication ethics. So these paraphrasing tools will help you in terms of rewriting something in your own words, right? So what are these tools available and how they are going to help you? That also I will discuss with you. And the AI tools which can help you in terms of finding the relevant journals for your research paper. Because one of the serious concern amongst the research scholar is you are not finding the relevant journal where you can publish your research. And there was one study which is conducted by US National Scientific Foundation, where they have reported that high impact factor journals, they accept only 10% of articles submitted for the publication. It means 90% of research paper submitted in various journals, they got rejected. And one of the important reason is we are not able to find the relevant journals for our research paper. You know, if you want to publish in Elsevier, if you want to publish in Taylor and Francis, Springer, Wiley, there are thousands of journals available. As per the UGC guidelines for the research scholar, there is a minimum condition that whatever research you are publishing, it should be UGC care listed journal, or it should be Scopus index journal. If you browse the journals indexed by Scopus, there are more than 46,000 journals are there. But you need one journal. You need few journals where you can publish your research. So these AI tools will help you in terms of identifying the right journal for your research paper. And almost all the publishers, they have designed and developed these tools. But the problem with the researcher is we are not aware about these tools. So what are these tools available and how you can access these tools, that also I am going to discuss with you. Then what are AI-based tools available which can help you in terms of detecting the plagiarism? You know, as per the UGC guidelines, anything which you want to publish, it should be below 10%. There are softwares available like Turnitin, Drillbit, Urkund. But now with the emergence of AI, now the students, they are taking help of AI tools in terms of writing their research paper. So what are the AI tools available at the same time which can help you in terms of detecting the plagiarism for your research paper? That also I am going to discuss with you. And you know, there is one report or there is one study which is published by Elsevier where they have reported that 50% of articles submitted in Elsevier journals, they got rejected because of what? Because of language problem. Specifically, researchers from developing country, whenever they are submitting their research paper in quality journals, there is a perception among the editor and reviewer that these researchers, they are not having command over English language. So what are the AI-based tools available which can help you in terms of improving the language part of your research paper? That is also very, very important, and I will discuss with you. And what are the reference management software? Because one of the important reasons behind plagiarism is we don't know how to cite right. Because if you are submitting your thesis, your research paper, there is a mandatory condition that all your references, it should be arranged according to any citation style. Suppose you want to arrange your research, all your references according to APA citation style. So there is a value of comma, full stop, semicolon, italics. So one thing is you invest your time. You spend 2-3 days just to arrange all these references. Another option is you use AI tools which can help you in terms of arranging all your references within few hours or within few minutes. So as a young researcher, you should know about these tools, and these tools will definitely make your research journey very comfortable, and you can submit your research or you can submit or publish your research paper within a limited time period. And the last thing is research data management. Now in medical sciences, if you are submitting your research paper to any journal, whatever you are reporting in your research paper, that is going to be supported with your raw data. Like for example, PLOS. PLOS is a very reputed journal in the field of medical sciences. So they have made it mandatory condition that unless you are not submitting your research data, they are not going to publish your research work. So what are these AI-based repositories are available, which can help you. Suppose you have selected one area. Now before going into that particular area, if you are interested in knowing about the research data, which is collected, compiled and analyzed by the researcher on that particular area, there are various data repositories available. So instead of going to your lab or going to the field, you can get the advanced knowledge about the data which is submitted by various researchers across the world. So what are these repositories available? That also I will discuss with you. Now, what are the tools available for literature review? Here, I'm not talking about Scopus or Web of Science, which are preferred tool for doing literature review. Here, I'm not talking about the library subscribed databases, because as I said, like in Punjab University, yearly we are spending 6 crore rupees. So you name any publisher database and we are having access of all these resources. But I'm not sure about this university. I'm not sure about the state university, how much funds they are spending in terms of subscribing to reputed database. But whatever tools I'm going to discuss with you, these all are free tools which is going to help you in terms of doing the literature review, whether you are a researcher, faculty or scientist. Like one of the very popular AI-based tool is Semantic Scholar. Now the advantage of this tool is, like for example, you can see from this slide, I just tried to search one area, drug discovery and development. You will find more than 53,000 articles published in quality journals. So this database is not only giving you result, it is also giving you in-depth literature review of any article for which you are interested. Like for example, these are the key benefits of searching through this particular database. It will immediately search for you paper summaries. You know, for researcher, one option is you download single paper, you just gone through with this paper, invest your 10 or 15 minutes just to read the abstract and summary of the paper. But with this database, immediately with the help of AI tool, it will create a summary of that particular research paper. That is going to help you in terms of taking a decision whether you want to include this paper in your literature review or not. At the same time, it will also give you related papers. Like for example, you are interested in one area. Now you have entered the keyword. When you will download 2 or 3 base paper, it will automatically create algorithm, and related to your base paper, it will give you a list of all the relevant papers which are indexed in this particular database, right? And it will also give you author profile. At the same time, it will also give you metrics and ranking. What is the citation of this particular research paper? There are 3 categories of citations. The influence of the paper on the academic community. So there are certain metrics which they have developed based on AI logic or AI algorithm, which is going to help you. Like for example, this is one example of the screenshot of one paper. You can see from here, this is the title. At the top, you will find all the bibliographical details of this paper. And below this, there are certain fields, like field. And then you will find another area, like what are the, like this, for example, from here you can see field of study. Like suppose you are entering one research area. So you will find research paper in different disciplines. Even you will find the same paper which is published by some social science researcher, keeping in view different perspective. So what you can do is, you enter the keyword and then just select field of study. And at the next column, you will find date range. Suppose you are interested in knowing about last 5-year publication on this particular topic. So from here, you can identify the date range and it will give you the result. Has PDF. Has PDF is an option where you will get access of all the research paper free of cost. Now you can ask me a question like, suppose Elsevier is publishing one research paper, how this website is giving PDF of the same paper? Because there is a concept of research visibility, where the researcher, they are making their research publicly accessible after checking all the copyright policies and research ethics. So there is a different concept. But when you click on this Has PDF, it will give you a list of papers where you can download free full text article of the paper. It will also give you list of top 10 authors who are working on that particular area. And at the same time, you will find other metrics also, right? So from this slide, you can see that Like for example, just now the first part is, this part is the concise summary of the paper, which is developed by the semantic scholar. So after reading this summary, you can take the decision whether this paper is relevant to your research field or not. At the same time, it will also give you abstract of the paper. It will give you total number of citation of that particular paper. It will give you some of the related paper. And the another advantage is you can chat with this PDF. For example, instead of reading the major findings, you can simply type in this chat box, what are the major findings of this research paper? Immediately it will give you major findings. What is the methodology of this paper? You can see there are predefined questions are there, but at the same time it is also giving you a chat box through which you can communicate with the PDF. So these all advantage you can get if you are searching your literature with this website or AI tool, which is called Semantic Scholar. I have very limited time, so it is not possible for me to explain you about each and every website. But here I am giving you overview. Rest of the thing you have to do. If you want to explore about the potential of these websites, whenever you are having free time, because these all are free resources. Right. Now talking about Elicit. Again, this is an AI tool which is going to help you in terms of doing the literature review within a very short span of time or within a few minutes. So these all are the benefits of Elicit which you can get while searching your literature. Like for example, this is the question. Now I want to search one topic, and the topic is, what is the impact of COVID-19 on school education in India? Suppose this is the topic. Now, what is the beauty of this platform is, now here you will find each and every segment of research paper in a tabular format. Right? And at the right side, you will find some other fields also. If you want to know about the variables, if you know about the limitations, if you want to know what are the sources they have referred while doing this study, each and everything you will see in this slide, it will display you in the tabular format. So based on this data, you can immediately take the decision, like what are the papers which I want to include in the literature review. The traditional method is you download the paper, you read the paper, and then you build the idea. The AI, the power of AI is everything it is doing on behalf of you. What you have to do is you have to just carefully screen, or you have to carefully go through with the data which is presented by this website. You will find each and everything about this paper in a tabular format, which can help you in terms of knowing what are the relevant papers. Now, another website is Consensus. Again, it's a very popular and AI-based tool, which is going to help you in terms of doing the literature review. Again, you will see that, suppose I'm searching on any research area. So whatever researcher they require while doing the literature review of any paper, most of the thing you will get with the click of mouse. You are talking about the methodology, you are talking about the population, you are talking about the variables. Each and everything is available in front of you. Right? What you have to do is, you have to explore about all these websites. And again, similar to other AI-based tool, you will find other options also, where you can get the literature review very easily and very promptly. Another website is Cite. This is for the faculty member. There's one thing about research is, again, there is a perception among the researcher. There's, as soon as you publish your article in Scopus Index or Web of Science Index journal, you will start receiving the citation, which is not true. Because as per this study conducted by Thomson Reuters, they have clearly mentioned that 43 SCI index paper, they have never received any citation. Right? And whatever citation you are getting from Scopus and Web of Science, it is only about the number. That your paper has been cited 100 times. Your paper has been cited 50 times. That's it. But what about the other details, citation details? Like how many researchers they have cited your findings where their study is in parallel with your study. What are the number of research papers where their finding and your finding is totally contrast? In the research paper, in which segment they have cited your paper, whether in the introduction part, methodology part, findings part, or conclusion part. Scopus and Web of Science, they are still working on these things. AI has already done it. So now many universities, because there is a pressure from NIRF, even in Punjab University, we are working on a project. I'm also part of that particular project, where we are giving each and everything about publication and citations. We are creating our own research platform, where we are taking help of these AI tools. So this website which will give you each and everything about the citation. Here you can see from this slide. This is the quick overview, right? And it will give you the result. Chat PDF, again, you all know about Chat GPT. I am not recommending Chat GPT for the researcher, because many publishers, they have clearly mentioned in their website, any article submitted by the researcher using Chat GPT, you are going to be banned for the lifetime. So don't ever try to use Chat GPT in terms of writing your research paper. But Chat PDF is available for you. Like for example, I have downloaded 10 papers for my literature review. But all these websites, they are not giving you any result about that particular PDF, because it is not possible for any website to index each and every publication. So suppose I have 10 papers. Now I wanted to know everything about that particular article. Go to Chat PDF, upload the paper, everything will get within a click of mouse. You want to interact with this research, you want to know about the findings, conclusion, abstract, summary, or you want to know about how they have developed the theoretical foundation of this research paper. Everything you will get with this website, which is very, very popular amongst the researchers. Even you can try this website, which is called ChatGPT. Research Rabbit, again, it's a very powerful AI tool which can give you scientometric data. It will connect all your research paper with the help of various indicators. Again, what you have to do is, for every researcher, whenever we are writing any research paper or we are writing PhD thesis, there are certain base papers or seed papers. You identify the seed papers, upload here, rest of the thing will be taken care about by the Research Rabbit. It will create you network of related paper. It will create network of similar author. It will create a network of funding agencies which are funding such type of projects or such type of research. So whatever you require related to your research funding, literature review, research writing, everything will get by this website, which is basically developed where they are using various metrics to build a network of some related papers. That is also very useful for you. Now, AI-based paraphrasing tool. As I said, if you are not aware about paraphrasing, if you have not developed the skills of rewriting something in your own words, then definitely you will indulge yourself in the act of plagiarism. Right? But there are certain tools are available. But again, I'm saying that I'm not advocating all these tools because sometimes they are not 100% reliable and accurate. But they will give you fair idea how these tools they are doing paraphrasing. And these are some of the very popular AI-based tools which can help you in terms of paraphrasing research. Like Quillboard, I think you are aware about the Grammarly. But the free version of Grammarly is giving you very limited option where you can use the potential of this software. But when you are subscribing Grammarly, then automatically it will improve the language of your research paper. But suppose if you are not having access of Grammarly, in that case, you use Quillboard, you use WordTune, you use Spin Rewriter. These are the tools which can help you in terms of doing paraphrasing. And this is one example, now the UGC is providing access to drill bit software to each and every university. Now your university, when you will submit your PhD thesis, they will check the plagiarism. So before coming for this lecture, I have done this homework. I have paraphrased 2 paragraphs of the original article with the help of Quillboard. So this is the paraphrased version, and this is the report of the software, where it is giving you 0%. That means no software is detecting the paraphrased version as a plagiarism. But the only thing which you have to very careful, whenever you are quoting, whenever you are paraphrasing, whenever you are summarizing, you have to give in-text citation of the original paper. Otherwise, it will be considered as a plagiarism. Specifically, it is called paraphrased plagiarism. Because you have taken the idea, but you are not giving credit to the original source. You can use these tools, but the only condition is you have to cite the original source after doing the paraphrasing. And when you will start using this tool, you know, your mind will automatically develop the algorithm, and it will tell you that in the next time, don't try to use these tools, because now you have understand the logic of doing the paraphrasing. Right? So all these tools are very popular, right? Now the next thing is finding the right journal. You know, sometimes the scientist and faculty member, they are also sometimes confused, because there are certain criteria through which we are selecting any journals. What is the impact factor, whether that journal is Scopus index is not, or Web of Science index, right? What are the aims, objective of that particular journal? Or sometimes, you know, we generally submit the article because other researchers, they are also publishing article in the same journal. But I think instead of following this traditional approach, what we can do is we can take the help of AI. Now, for example, suppose I want to publish my article in Elsevier journal. Now, Elsevier has developed a powerful AI tool which is called Elsevier Journal Finder. What you have to do is you simply copy and paste your title, abstract, keywords, and your area of study or your field of specialization. Again, this platform will only work if you are writing the abstract in a structured manner. You know, again, there is a lack of awareness about the researcher how to write a good abstract. Any good abstract will definitely include these 5 or 6 components. And what are these 5, 6 components? First is, what is the purpose of this research? What is the main objective of your research? What are the major findings? What is the methodology which you have applied while doing your research? What is the limitation? Is there any social implication of your research? Is there any practical implication of your research? Then only it will be considered as a quality abstract. So suppose if you are writing the abstract in such a manner, in that case, this platform is going to help you in terms of identifying the journals. Like for example, this is the title, abstract, keyword, and field of my research. Now here it is giving me list of journals, and at the same time, it is also telling me what are the chances of publication of my article in these journals. So you will see there are few very rare journals, or there is very less chance of publishing my research in Elsevier, because Elsevier is not publishing such type of research, right? So high chance of acceptance of my research paper, right? So likewise you can do this thing, right? Then Springer is also giving you platform where you can do. Idange is a very reputed platform which can help you, right? Then Jane, journal author name suggester. Then EndNote Match if you want to publish your article in SCI index journals. So that is going to help you in terms of selecting the journal. If you want to publish in IEEE, IEEE is also, so almost all the publishers, they have developed the platform. Now talking about the tools, these are some of the tools which can help you in terms of detecting the plagiarism. These all are very popular and very powerful AI tools, because now I got the slip where I have to complete my presentation within 5 to 10 minutes. So I'm just, giving you a quick overview. I'm not explaining you, but what you can do is you can always use these tools, right? Then this is the screenshot like copy leaks. Now AI-based grammar checker tools, like Trinka. Trinka is very, very popular and powerful tool. You can see the example. You upload your paper and all the grammatical and language part, it will reflect you at the right-hand side. So you can see whether it is giving you correct suggestions, or if you don't want to accept the changes, that also you can do. But anyways, these tools will help you almost 50 to 60% in terms of improving the language part of your research paper. These are some other tools like Hemingway Editor, Silkwrite. These all are AI tools which can help you in terms of improving the language part. Now talking about managing all your references, I think some of you might be aware about these tools like Mendeley. I think in Punjab University, I am delivering lecture in pre-PhD coursework or workshop or an invited lecture where I am giving practical aspect of how you can use these AI tools, like how you can integrate your laptop with Mendeley software so that all the references will be arranged according to any citation style, like EndNote, RefWork, Zotero, all these tools are. Now talking about managing your data, this is one of the example of Elsevier paper, and the first reference is not about any research paper, conference paper, book chapter, or any other review article. The first reference is about this data, right? About this data which is uploaded on this AI tool which is called Figshare. Now Figshare is a data repository where the researchers, millions of researchers, they are uploading their scientific data. What is the advantage? The advantage is paper will give you citation for your research paper. If you are uploading your data in these repository, you will get the data citation. Because every data is having its own interpretation. So it is not like that you are the best person who have given the best explanation of your data. There is always a possibility that someone else can explain your data in a much better way. So that is why researchers all over the world, they are uploading their scientific data so that other researchers, they can also use these data for giving the explanation or interpretation of the result, right? So just to conclude my presentation, what I am trying to say is, we have to use these tools. I am not saying that we have to make 100% dependable on these tools, but these are research assistive tools. And if you are aware about these tools, if you know how to use these tools during your different stages of your research, that is going to make your research journey more comfortable, and you can do research in a timely manner, right? So with this word, so thank you all for inviting me, and thank you for listening to me.
So, Dr. Neeraj Kumar, it's a very informative lecture, and I think not only the students, but most of the faculty members also got benefited. And you have very beautifully explained from first steps for selection of the problem to submission of the thesis, including all the journey of the PhD or any M.Pharm, that how to select, how to do, how to write, how to publish, how to select the correct journal, and how to get your things done in a proper time. I always ask whenever in any RDC that what is your plan, and 90% students do not know what is their destiny.
True.
That how to plan their research, how much time they required, how many facilities they required, in which journal they want to publish, what is the authenticate source of the impact factor of that journal, how to check the impact factor of journal, how to know the citation index.
Yes.
And whenever they are going for any defense, these all question is going to be asked.
Yes.
For that probably they have not been trained and they have not knowing it.
Yes.
So sir, very nice lecture, and I want that in every research methodology subject that M.Pharm is also having, a PhD is also having.
Yes, sir.
Person like you must give the lecture. Thank you very much.
Thank
Now
you.
this session is open for question answer, and I will give my this mic to my co-host also. He will also want to comment on.
Yes, any query to the sir, because it is a very relevant topic. I think an eye-opening topic, sir. You have
Thank you,
provided.
sir. Thank you.
really, in a very simple, yeah, you have a query?
Thank you so much, sir, for your great insights on all those AI tools. Everyone who was attending this lecture, and even it was a fourth lecture in this, morning session, and nobody was feeling hectic because, and right from the seniors to the juniors, everyone was clicking the pics for their, record. It seems that it was very great and insightful. Sir, I have two questions actually. When it comes to the artificial intelligence and including the AI tools for your research and academia, so one most important point is as the title, first point of title is research ethics.
Right.
So what is the ethical aspects of using AI tools in research and academics? Because even though if we are giving the insights on this, we should also take that part into the considerations as well. Okay, what should be the responsibility of any student? What should be the responsibility of any research scholar? What should be the responsibility of any academic institution? And what should be the responsibility of any editorial journal or any publisher regarding the using of any AI tool for the research and academics, which is a very, very important aspect. This is my first question.
Yes, yes. Yeah, okay, talking about this research ethics part. So for young researcher, you have to understand in a very simpler manner, research ethics, or you can say the publication ethics or research ethics is having three components. Plagiarism is one of the component. Other two components are falsification and fabrication. You know what is falsification and fabrication? Where you are reporting your data falsely. Like for example, in your research methodology, you have mentioned that in my research, I am going to take a sample size of 300 respondents. So instead of taking the response from 300 respondents, what you are doing, you are collecting the response from only 150, rest of the thing you are manipulating. There is no software in this world which can detect this sort of fabrication. What is falsification? That you are writing in your paper that you have followed the mixed approach while reporting the data or while collecting the data. That is, you are going to the field, you are making the observation, you are doing the interviews also. But you are reporting the observations which you have never made. So this is called falsification. Plagiarism is when you are taking someone else's idea, thought, text, data, images, anything which is published by some other researcher. So now talking about the role of AI tool, whatever tools I have explained to you, you talk about the literature review. Literature review tools is basically to give you insight about the literature. You are not directly copying anything from these tools. The only tools which is directly related to the research ethics is called paraphrasing tools. I've already mentioned you that there is no harm if you are using these paraphrasing tools. But you have to be very, very careful because sometimes these tools makes arth ka anarth. You know, they will change the meaning. So you have to carefully read the paraphrased version, and if you are satisfied with the paraphrased version, or you have incorporated your inputs also in the paraphrased version, the only thing which you have to remember is you have to give in-text citation. Whenever you are giving in-text citation, it means you are telling the reader of your paper that this portion of work is not yours. This you have taken from this source. Sometime reader of your paper, they are more interested in reading the original article rather than your paper. So if you are giving proper acknowledgement, then you are 100% safe. Now you have also asked me the responsibility of author, editor, researcher, peer reviewer. That is why all the quality journals, they are bound with COPE guidelines. Go to Google and just search COPE guidelines for research ethics. They have clearly explained each and every responsibilities of different stakeholders who are involved in the research, right? So there is no harm of using these tools, but at the same time, you have to give your own input, and wherever you are thinking of giving credit to someone else's work, give credit to already published sources, right? What is your next question?
Sir, my next question was that when it comes to detection of any plagiarized work, So right now, apart from the plagiarism report, whether you are taking from any software, either Turnitin or any other, you also have to submit one AI report as well, along with the plagiarism report.
Yes.
But when we are saying the plagiarism, percentage, as per the UGC, it should be less than 10%.
Yes.
But because maximum institutions as well, right now, they are asking for the AI report as well, along
Yes.
with the
Yes.
plagiarism. But there is not any, percentage limit, because maximum universities are allowing for the 20%. But what's about their regulations? They
Yes.
are not making any, some limitations of it. They are not making any stringent limits for that as well.
Yes.
So what's about that? What regulation should, go forward for it?
Yes, I think almost all the universities in India, they are following UGC 2018 regulation, which is clearly about the plagiarism or research ethics. You have asked the question about AI generated content. Now, UGC has given access to all the academic institutions of the software, and the name of software is Drillbit. So Drillbit is generating the report where you are using, suppose if you have copied something which is from AI generated tool. At the same time, it will tell you, like, if the percentage of your paper is 16%, it will tell you what is the percentage from AI sources. Even the Turnitin, they have incorporated this feature. Suppose in case you want to be 100% sure whether this work has been copied from any AI tool or researcher has used any AI tool in terms of generating the content. So in my lecture, I've given you name of some of the tools which are specifically meant to check the similarity through AI generated tools. Like for example, how these softwares, they are checking the AI tools, AI generated content. You have to understand the logic, how this software basically works. Why Turnitin is very popular? Because Turnitin is having bigger repository as compared to any other anti-plagiarism software. So suppose you are submitting your paper, immediately it is checking the plagiarism from millions of paper. But all these AI tools like ChatGPT, Google Bard, Turnitin or any softwares, they are not taking their content into their repository. Now the question is how they are working. They are recognizing the pattern. Because all these AI tools, they are basically writing the content on a similar pattern. So these tools basically recognizing the pattern, and based on the pattern, they are telling you that there are chances that this portion of your work you have created with the help of AI tools. So Turnitin, Drillbit, both these softwares, they are giving you AI report also, right?
Thank you so much, sir. And I just add to one more point here that first point was, AI tool for literature review, because for every research scholar, it is a hectic part of their research.
Yes.
And apart from, as you mentioned, that semantic scholar site, so I will suggest few more. There is a one, that is called connected papers.
Yes.
Which is very good, database for you just have to download one paper, keep it, that paper should be your seed paper. And all the papers who are connected will be, displayed just like the seed, site one. Yes. There is another database that is called lit maps, that is also working in the same way.
Yes.
One, there is Perplexity. This is another data, AI based for literature review. System Pro, many more. You can explore. And I just want to add one thing, sir, because you gave the presentation, because for this type of talks, if we will get the hands-on training, a workshop, that would be much more insightful. Apart from this portion, because I am attending such type of events. They are dividing the events in two parts. First one is just the theoretical base. Another portion they are doing, sir, hands-on training, so that reviewers can get more in depth into it. Thank you so much, sir. We will disconnect you in future as well.
Thank you. Thank you.
Any
Thank
other
you so
student?
much.
You want?
So firstly, thank you for the information. And my question is, as you said, the plagiarism is a most important part of any reviewer research paper. And if you are saying that ChatGPT, Perplexity, Gemini, Bard, these artificial intelligence tools, or I can say large language models, cannot be used for this purpose, then I want to ask that if I train a large language model or an AI model that is on my local server, the API of that particular AI tool is not available on the internet, then is it possible for anyone to detect my flags?
I think I hope I'm audible. You know, I can give you a quick overview of all these tools, but why I'm not advocating any AI tool for writing your research paper is because it will finish your logical thinking. You will become so much dependable on these tools, you will never try to understand about the research. As a young researcher, what you have to do is, you have to make use of these tools, but you have to, think from your own perspective. If you are not having knowledge of research methodology, if you are not aware about the research design, how you can write a good synopsis, which is a blueprint for your research. In Punjab University, like almost in all the departments, as previous student has already mentioned, that I am giving hands-on sessions, not only about these tools. There is a software which is called LaTeX. Now I am encouraging research scholar, even the faculty member in Punjab University, why you are not insisting research scholar to use LaTeX, right? And the beauty of LaTeX is you only focus about the content. Rest of the thing will be taken care by LaTeX. There is no need to prepare table of content separately. List of tables, list of figures, acknowledgement, whatever preliminary pages you are preparing while submitting your PhD thesis, that is already taken care by LaTeX. You only have to understand the simple basic HTML coding. You can watch any YouTube video and you can install the LaTeX software and you just play with this LaTeX. Now, one last thing which I want to share here is, sometimes we receive the desk rejection of our paper, and the reason is that we have not submitted our research paper according to the publisher's template. Like for example, if I want to publish my article in IEEE, if I am simply submitting the Word file, next day I will receive the desk rejection. And what is desk rejection? Desk rejection is a slap on the face of the researcher that you don't know, you know nothing about research. So what is the beauty of LaTeX? You only type the content. There are already 600 templates are there. You select any publisher template. All your research will be arranged automatically according to that template. So as a young researcher, you have to invest your time. You have to learn about all these tools. And definitely these tools are going to make your research journey more comfortable. You will be able to complete your research within 3 years. Right? But don't try to use these AI tools in terms of writing.
Yes, sir.
This is my own issue.
Okay, sir. Thank you.
So simply, sir, we can use as a tool. It's a no substitute for your own research, your own idea. Because what we feel in last 1.5 years, that now students are depend on that. So that logical thinking, analytical skills, interpretation of data, that are missing. And if you do not have the base, these tools are not going to be help you, anything. Topic is the most important, research is most important. These tools will help you how to write, how to present. This is just for a presentation purpose. So use it as a tool, not as a just a substitutes for everything. This is, thank you. Thank you very much.
Thank you so much, sir, for sharing your knowledge and experience such amazingly how to use AI tools, how to integrate it, the apps and tools. I'm pretty sure that all of us who are sitting here, this session was much helpful, especially to our budding researchers. A big thank you to one of our attendees as well who enhanced in our knowledge and gave the suggestions. Although I don't know the name, we really appreciate your suggestion as well. I kindly request our chair session members to present a token of appreciation to our worthy speaker, Dr. Neeraj Kumar Singh. I request Dr. Subhit Jain and Dr. Suresh Thakur to join us on the stage. Thank you so much, dignitaries. Now, ladies and gentlemen, going from national to international, I'm privileged to inform our audience that our next speaker is Dr. Sudha Shankar from Tel Aviv University, Israel, connecting to us virtually. I request our media team to connect us to our worthy speaker, Dr. Sudha Shankar. I kindly request Dr. Manisha Bhatia to present the welcome sapling to Dr. Sudha Shankar, and I do request Dr. Seema Bansal to receive the sapling on behalf of our speaker, Dr. Sudha Shankar.
Thank you very much, ma'am, for joining us, though through online mode, but we are really overwhelmed by your pious presence.
Thank you so much for the kind introduction. I hope everyone is able to see my screen. I am trying to share it.
To the audience, I'd like to inform that Dr. Sudha Shankar is currently working with Professor Hud Gazit in the Medicinal Chemistry Unit of Blavatnik Center of Drug Discovery as postdoctoral fellow. Her current research interests involve in design and synthesis of novel molecules, resolving challenging synthetic problems, and ADME, SAR analysis. Also, her work includes synthesis of peptides and to explore aggregation propensity. And she is awarded for prestigious Newton International Postdoctoral Fellowship by Royal Society and Academy of Medical Sciences, UK, to work on total synthesis of botryomycin and their analogues using solid phase peptide synthesis at School of Chemistry, University of Glasgow, Scotland, UK, with Professor Andrew Jamieson. I now hand over this session to Dr. Sudha Shankar. Ma'am, please begin with your presentation. Thank you.
I will share my presentation. Sorry. Sorry, I'm trying to share my presentation, but I'm not able to see the option where I can share the screen. So you are able to see my screen or my presentation? Anyone can help me to just validate what you can see on the screen.
ये नहीं कह रहे क्रोम में मिरर एक्सटेंशन डाउन
Wait. presentation. First, my voice is audible?
Dr. Sudha, your voice is audible.
Okay, thank you so much for the confirmation. Sorry, yeah. So before starting my presentation, I would like to thank for this kind invitation from the Professor Sumit Gupta, Principal of College of Pharmacy, and Dr. Nidhi Gupta, Assistant Professor, College of Pharmacy. And I did my PhD with Dr. Nidhi Gupta, and we know each other from long time. And it's my great pleasure and honor to be the part of this gathering. And as this conference is all about the research ethics and clinical therapeutics and the phytopharmaceutical drug, I'm going to discuss about the peptide-based drug discovery, which is also a part of phytopharmaceutical drug, because all of us knows that peptides, some of the peptides are well-known phytopharmaceutical drugs. And before starting my presentation, I would like to give a very brief introduction about the drug discovery process. So this drug discovery process starts with the millions of compounds and it ends with the single molecule. And there is a lots of levels to screen the molecules, and to perform this drug discovery process, we need the expertise from the different field, from the medicinal chemistry, from the biology, from the computational chemist, and we need expertise, and we need lots of money to make a single drug, marketed FDA approved drug. And this whole drug discovery process, it takes 10 to 15 years of time, and the process starts with the identification of the therapeutic target of the interest, and it includes assay development method, lead identification, characterization, formulation, and animal pharmacological activities, pharmacokinetic activities, and safety testing on the animals and also on the human in the clinical studies. For the target identification, what we are doing at our center, uh, we are working to find a novel target, and that target depends on the disease of the interest and based on the medical need. And we need some tools and technology to identify those targets, and we have the knowledge of the molecular mechanism behind that particular disease or the target. And the target identification, we are using different approaches like affinity based approaches, phenotypic based approaches, genetic approaches, and the computational approaches. And so once we find our target, we have to validate our target, and that is called the target validation in this drug discovery process. And that target is kind of proteins, enzymes, and the signaling receptor which is present inside the cells. And that the target should be good, efficacious, and it meet the safety need and the commercial needs. And when we are doing the target validation, we have to keep in mind some important criteria for the target validation, like genetic manipulation, antibodies, and some chemical genomics. And once we know our target, we validate the target, then we move to the screening of the molecules. And maybe we are thinking from where these millions of molecules are coming from, because these molecules have a huge library, like all of know about the Cambridge library, which is one of the biggest library where from where there we can take the molecules. And also the another source is the natural product libraries. There are libraries based on the computational and medicinal chemistry, or using the information from the existing molecule with the known bioactivity. And once we get our molecules, we have to like identify what are the best molecule, because we have a millions of molecules, so we can't take all the molecules directly for the cell based assay. So for that, we perform in silico pre-screening of the molecules, and that in silico is called the computational study. It's a computer simulated screening of structurally related molecules. And this helps in silico screening, helps in finding the structure that are most likely to bind to the target, optimization of the target affinity and the selectivity, and also this in silico screening. And there are several medicinal chemistry tool nowadays exist from where we can predict the absorption, distribution, metabolism, excretion, and the toxicity properties of the molecule. And by these tools, we filter most of the molecules at the initial stages in order to reduce the toxicity and to eliminate the side effects. And this in silico pre-screening is much more economical than the high throughput screening. And this high throughput screening, what it means, like we in our center, we are performing this high throughput screening unit using a robotic system. And this robotic system, at a time, it is screened like millions of compound for the different targets. And once we perform this high throughput screening, we find our best hit molecule. And at this stage of high throughput screening, it's a kind of go no go criteria. We can decide what are the molecules which can proceed further for the drug development, and what are the molecules we can discard. And there are two different assay performed in the high throughput screening. One is the primary assay and another is the secondary assay. Primary assay is the single concentration based assay, and the secondary assay, it depends on the concentration dependent, and where we can calculate the IC50. But the most of the compound in this high throughput screening has a negligible potency, and only small fraction. And based on my experience, we found that only 1% compounds show a promising result uh, at this stage. And once we get our hit molecule from the high throughput screening unit, further we move to the hit identification, lead discovery, lead optimization, and this is called hit to lead optimization of the molecules. And this lead optimization process, it mostly strives to improve the potency of the molecule against the different target protein in order to improve the ADME property and their binding affinity, and in order to find a novel molecule which can be patentable further. But theoretically it's look like it's just a next step. But choosing the best molecule is not a straightforward process. It needs lots of expertise, lots of time, lots of money. And this is the whole process of drug discovery. And this drug discovery process, it's same process for either it's a peptide drug, either it's a small molecule, some biologics, and some carbohydrate based molecule, etc. But today in lecture, my focus is on the peptide based drug discovery. And before like going to the peptide drug development, all of us know about what are the peptides. These peptides are present in all the living system, and the proteins are made up of peptides. And these proteins are made up of polypeptides, but still there are some peptides exist in the living organism, and they act as a signaling molecule in the body. And that's the reason the peptide drug discovery get like more popular, like what are the effect of peptides. And these peptides are also not only applicable in the field of drug discovery, but also for the structural biology, vaccine development, tissue engineering, where most maybe you are familiar with the hydrogel based tissue engineering wound healing assay, and also as a drug delivery agent. And so I'm going to talk about how the peptide drug development is started. And the first peptide drug is started in 1922, where we use first time the insulin peptide drug, which is extracted from the animal source. And it's 1922. So last to last year, we celebrated this 100 years of the insulin as a marketed drug. But there is some limitation in the peptide drug, and that's the reason the peptide drugs are not easy to synthesizable and to become a marketed drug. And the most an earlier time, most limitation was with the synthesis of these peptides. But in 1963, the discovery of solid phase synthesis by Merrifield, it create more interest for the peptide based drug discovery. And nowadays it's becoming more and more popular, and there are new technology evolved so far to develop like phase display, recombinant technology, and many more new chemical approaches. And this year also in 2023, there are five peptide drugs approved by the FDA among 35 other FDA approved drugs. And if I will talk about the current market landscape of the peptide drug, only 66% are the peptide drug exist in the market till date. Still, the small molecules are in more quantity. But, however, only the 16% of this peptide drug covers 45 billion US dollar of the market, of the whole drug market. And I can't share my screen, but I want to show that how the peptide drug evolved, and I have the data from 2010 to 2023, and based on the annual growth rate of the peptide drugs from 2010 to 2023, scientists are predicting that by the 2033, it reaches from 45 billion US dollar to 106 billion US dollar. And the interest for these peptide drugs, because these peptides are the best of two different worlds, and the other two different world is the small molecule and the biologics. The small, it means the peptides combines the advantage of both the small molecules and the biological like antibodies based drugs. And the advantages of the peptide are their high specificity, good efficacy, good safety because they are more biocompatible than the other molecules, low immunogenicity and membrane permeability, and low cost. And if I will talk how we start the process of drug discovery in the peptide, so we all knows that in our body every disease has some reason, and the reason it starts from the protein-protein interactions. So whenever we are trying to focus, for example, we are trying to develop an anti-cancer drug, so we have to find the structure of that particular protein which is responsible for that particular disease. And the protein-protein interaction, there is a common like boundaries between two different protein which help in the interaction, and it causes like cell damage. uh, it's unwanted cell growth. So in order to find which is the fragment responsible for the interacting of two different proteins, we try to extract that fragment. And once we get the extracted fragment of that protein, we optimize that protein, optimize the binding affinity of that protein with the another protein by the computational study. And then we perform the epitope mapping of that protein because it's a long chain of amino acids. So we try to find what is the most important binding site in that protein. And based on the sequence, we do further modification and we make a peptide library. And there are different types of peptide library like overlapping peptide library, truncation peptide library, random peptide library, alanine scan, positional scanning, and scrambled peptide library. And from that library, we screen those peptides for the biological activity, and further we optimize our peptide. And it's very important to optimize. This is the most challenging part in the peptide drug discovery because peptides has lots of challenges as compared to the small molecules because of the bigger size, and it originates from the biological system. So there is a chances that it can show some like other effects inside the body. And the some challenges are like poor bioavailability, oral bioavailability, proteolytic stability, and some synthetic challenges and short half life. And in order to overcome all these approaches, we have some techniques and tools and some strategies to overcome these challenges. Like if we have a linear peptide, we can try to make a cyclic peptide, which is more stable than the linear peptide. We can do terminal protection like C-terminal protection, N-terminal protection in the peptide. Also, there is another technique to find which is the crucial residue which involved in the protein interaction. And once we find the crucial residue, we try to remove the other unwanted residue. And another approach is the computational modification in the peptide. And the next one is the backbone modification. In backbone modification, we use beta, gamma, different kind of unnatural amino acids. And there is the last one, it's a side chain modification. But among all these issues, the peptide stability is the key important points to consider for the peptide designing. And that I'm going to, if peptide is going to use in vivo, in vitro assays, no matter how what best peptide you design, but if peptide is not active at your target site, it's not going to be useful anymore. So when we design when we consider this peptide stability, we have to keep in mind two main points. First, how the peptide will behave if we are doing the biological assay, and how the peptide will behave during the synthesis process. Because during the synthesis process, there are chances of oxidation, hydrolysis, and the deletion sequences. And during the peptide assay, there is a chances of protease degradation, renal clearance, etc. So, in order to overcome these, in order to make a more stable peptide, we have some advanced design techniques like I'm not able to share my screen, but I will tell. Like there is a chances, like if you are designing your peptide and there is a glutamate at the end terminal of your peptide, there is a chances of cyclization. So in that case, we decided, okay, we can replace this glutamate to some other amino acid, or either we can acetylate that glutamate, because the glutamate easily gets converted into the pyroglutamate, which is not the native structure of the glutamate amino acid. Similarly, if there is a aspartate and glycine residue together, it again gets cyclized. Another issue is with the secondary structure formation. If we have a multiple glutamine, isoleucine, leucine, phenylalanine, tyrosine, valine in our sequence, there is a chances that it leads to the formation of secondary structure. So we try to replace these. For example, we have a glutamine, we can replace with aspartate. If we have a threonine, we can replace with the serine, because they have a similar kind of electron density around that amino acid. So it retains the secondary structure of the peptide. Similarly, there is a chances of hydrolysis, oxidation if we have methionine, cysteine. So we replace cysteine with serine, methionine with norleucine. Similarly, in case of hydrolysis, we can, like if we have aspartate and glycine, aspartate and proline, serine, then we can replace the aspartate with some other amino acid. And next, if we want to make our peptide more stable for the proteases, there are some strategies like cyclization, acetylation, amidation, D-amino acid replacement, peptides, and hydrocarbon stapling. There is a report in Nature Biotechnology. It's a recent report, and in that paper, they reported that it's a case study I want to discuss. So, in that, they developed a analog of PTH hormone, parathyroid hormone, and in that parathyroid hormone, what they did, they removed the alpha amino acid with the beta residue. Beta residue means that beta amino acid is different from the alpha amino acid with the extra side with the extra CH group, and because of that, replacing this alpha amino acid with beta amino acid, it increases the half life of the amino acid, and in that case, it's not going to have a very quick renal clearance from the body, and you don't have to take frequent medication. And after introducing this beta amino acid, we improve and make more potent molecule, PTH molecule, than the native PTH. And another study, it's reported with the D amino acid replacements when we replace L amino acid with the D amino acid. As we know, most of our body, most of the proteins are made up of L amino acid, which are the natural amino acid. For but when we replace with the D amino acid, for example, there is a drug called vasopressin. It's a very well-known drug, and for the blood pressure regulation. And in that case, they develop an analog of vasopressin, and they replace the L-arginine with the D-arginine. And in that case, we increase the half-life from 10 to 35 minutes to 3.7 hours. And there are some other example of anti-arrhythmic drug, Ebola peptide drugs, HIV peptide drugs, where we replace all the L amino acid with the D amino acid, and those kind of peptides are known as the mirror image peptides. And they are also a very popular trends going on nowadays in the peptide-based drug development. And other point we have to keep in mind for the peptide stability is the renal clearance of the hydrophilic peptides. So if we have a hydrophilic peptide which molecular weight is less than 25 kilodalton, they are they are much easily filtered through our kidney, but once it get filtered, it's for peptides is very hard to reabsorb from the renal tubule. So in order to reduce the renal clearance time, we have some techniques like we can conjugate those peptides with some polymers like polyethylene glycol, polycylic acid, hydroxyethyl starch, and bovine serum albumin. These are some common approaches people are using in order to improve the renal clearance of those peptides. And apart from that, nowadays, very new technologies are coming in the field of peptide discovery, and the most common is the display technique. Display technique, like it started with the phase display technique of those peptides, and nowadays, it's the most popular is the mRNA display screening of those peptides. And in this mRNA display technique, it's used to develop a large library of the peptides and proteins with the high affinity of a specific target. And here, what scientists are doing, they're taking the DNA template and they converted that DNA template into a mRNA library. And we know the process of translation and the transcription, it happens in our body as well. And the same process they are doing in vitro. And in this, they make the library of mRNA, and that mRNA is attached with the puromycin, and that puromycin helps in the translation process. And once the mRNA started synthesizing the peptide, it synthesized a nascent peptide, and that peptide transferred to the puromycin A side of the chain, and then the reverse transcription process happened. And once we get our peptide, we try to find a good target for that peptide. And there is a process when we find the good target for that peptide, then again that mRNA it get converted into the DNA. And if we find a good target, that DNA again get amplified and transcribed for the next round of the screening. So this is the RNA display technique going on. And so I will end up my presentation here with some conclusion for the peptide drug discovery, because it's becoming more and more popular nowadays as a therapeutic class. But the design is very challenging. The design and the purification of these peptides are very challenging. But in order to overcome these challenges, we are sure that the peptide drug, based drug will be accepted as a genuine alternative for the traditional molecule therapeutics. And peptide-based drug approaches are the emerging as a valuable tools for addressing several unresolved disease area. Thank you so much for your kind attention. And sorry I was not able to share my presentation, but still I am happy to take some questions.
Thank you so much, ma'am, to deliver such a wonderful lecture and to enhance our knowledge about the topic, and especially for connecting with us.
Thank you.
For the felicitation ceremony of the speaker, I would request Dr. Sarita Sharma to please come up on the stage and appreciate the speaker. I do request Dr. Nidhi to come up on the stage and take the memento on the behalf of speaker, Dr. Sudha Sharma. Thank you so much, dignitaries. We will continue the event after lunch with a valedictory function. You all are requested to have your lunch.
Organized by College of Pharmacy of Nursing, Mulana Ambala. Ladies and gentlemen, we'll be proceeding towards the valedictory ceremony of the day. But before that, we would like to felicitate our resource persons. For the same, I would like to call upon stage Dr. Jitendra Singh, Principal IET Badal, to join us on the stage. And I would like to call Dr. Sandeep Kumar Ranga in order to present the token of appreciation to Dr. Jitendra Singh. Please join us on the stage. Thank you so much. I'd like to request Dr. Vinod Gautam to join us on the stage. For the same, I would like to invite Dr. Sunana to please present the token of appreciation to our resource person. Thank you so much. I cordially invite Dr. Pradeep Goyal to please join us on the stage. And for the same, I request Dr. Ishrat Zahoor to present a token of appreciation to them. Thank you so much. I request Dr. Kavita Munjal, one of the resource persons, to join us on the stage. And for the same, I cordially invite Dr. Anuj Malik to present the token of appreciation to our resource person. Thank you so much. Ladies and gentlemen, we'll be beginning with the valedictory ceremony. For the same, first we'll be having the category of poster presentation from Shodotsav 2024. We have 3 prizes ranging from first to third, and the third prize winner under the category of poster presentation is Dr. Harish Sharma, a PhD scholar from MMDU. Many, many congratulations. The second prize winner is Miss Isha Chawla, again PhD scholar from MMDU. If they are present, please join us. And ladies and gentlemen, the first prize winner under the category of poster presentation in Shodhotsav 2024 is Dr. Tarun, again a PhD scholar from Maharishi Markandeshwar deemed to be university. Many, many congratulations to all of them. Proceeding forward, we will be having the prize winners under the different categories of poster presentation, starting from the pharmaceutical chemistry. So the third prize winner in the category of pharmaceutical chemistry is Hamida Bano from School of Pharmacy, Deshbhakt University, Mandi Gobindgarh, Punjab. Many, many congratulations. The second prize winner is Vaneet Kumar from Chandigarh College of Pharmacy, Chandigarh Group of College, Landran, Mohali, Punjab. And the first prize winner is Riya Singla from Devilal College of Pharmacy. Many, many congratulations to all of them who have bagged the positions in the category of poster presentation under pharmaceutical chemistry. So these members will be getting their prizes and the certification on the front desk. I request Hamida Bano to please come on the stage and join me here. Miss Hamida Bano. She has bagged the third position under the category of poster presentation in pharmaceutical chemistry. Congratulations. I request Dean and Principal, Pharmacy College, Dr. Sumit Gupta to present the token of appreciation to Hamida Bano from Deshbhakt University who has bagged third position under pharmaceutical chemistry. Sir, aap present kar dijiye, please. We would love if you will present the token of appreciation. If some of the faculty from the said university are present here, please join us on the stage from Deshbhakt University. Congratulations. The second prize winner, Vineet Kumar. I request them please step forward and join us on the stage. Vineet Kumar from Chandigarh Group of College, Landran, Mohali, Punjab. And the first prize winner, Riya Singla from Devilal College of Pharmacy, Boria. Please join us on the stage. So Proceeding forward towards the second category, that is under the pharmaceutics subject, we have the third prize winner, Aditi Kumari from Department of Pharmaceutics, College of Pharmacy. Congratulations. The second prize winner under pharmaceutics subject is Akash Chandel from Chandigarh Group of Pharmacy, Landran Mohali. Congratulations, Akash Chandel from Chandigarh College of Pharmacy, Landran Mohali. And the first prize winner, Muskan from School of Pharmacy, Deshbhakt University, Mandi, Gobindgarh, Punjab. Muskan from School of Pharmacy, DBU. Congratulations. These were the prize winners under the category of pharmaceutics. Now coming to the next category, that is pharmacy practice. We have the third prize winner, Sumit Chaudhary from Department of Pharmacy Practice, CT Institute of Pharmaceutical Science, Shahpur, Jalandhar. Sumit Chaudhary. The second prize winner is Aarti Saini from College of Pharmacy, Maharishi Markandeshwar deemed to be University. Aarti Saini is the second prize winner under pharmacy practice. These are the prize winners under poster presentation in different categories. And the first prize winner under pharmacy practice is Khushbu Baranwal from Department of Pharmacy Practice, National Institute of Pharmacy Education and Research, SAS Nagar. Many, many congratulations. First prize winner is Khushbu Baranwal. Proceeding forward, now under the category of pharmacology, the third prize winner is Simran from College of Pharmacy, Maharishi Markandeshwar deemed to be University. Congratulations. And the second prize winner is Rohit Kumar from Aman Pharmacy College, Rajasthan. Congratulations. Rohit Kumar is the second prize winner under pharmacology in the category of poster presentation. The first prize winner from pharmacology is Sujata Singh from College of Pharmacy, Maharishi Markandeshwar deemed to be University. Many, many congratulations. Sujata Singh is the first prize winner in pharmacology under the category of poster presentation.
For the awards under pharmacognosy category, we have third prize is backed by Monica Sharma, the School of Pharmacy, IEC University, Baddi, Himachal Pradesh. I repeat, third prize under pharmacognosy category is backed by Monica Sharma. The second prize is backed by Aishna Kalra from College of Pharmacy. The first prize under pharmacognosy category is backed by Shiza Ashraf Malik from School of Pharmacy, Mandi, Gobindgarh, Punjab. Congratulations to all the winners.
Proceeding forward, we'll have the winners from the another category, that is oral presentations. Ladies and gentlemen, there are a number of students who have bagged the second position under the said category. For the same, the first one who has got the second position under oral presentation is Vanshika from DIT Dehradun. She has done it through online mode. Congratulations. And another one is Dr. Aisha Siddiq from King Khalid University, Saudi Arabia, again through online mode. Congratulations, she's an international attendee. And another one is Ranjot Jeet Singh from Department of Pharmacology, Institute of Medical Science and Research, Maharishi Markandeshwar, deemed to be university. Another one is Miss Annu Kaushal from Medical Surgical Nursing Department, College of Nursing, again from Maharishi Markandeshwar, deemed to be university. Many, many congrats. And another participant who has bagged the second position is Dr. Swarna from College of Pharmacy, MMDU. Congratulations to all of them who have bagged the second position under the category of oral presentation. So a total of 5 candidates are there who are gained the second position under the category of oral presentation. Dr. Swarna, Ms. Annu Kaushal, Ranjotjeet Singh, Dr. Aisha Siddiq and Ms. Vanshika. So for the same category, the first prize winners are number 1, Ritu Tomar from DIT Dehradun, again through online mode. And another is Sabina Yasmin from King Khalid University, Saudi Arabia, which is through online mode. And another one candidate, Ms. Vanshika from Department of Pharmacology, College of Pharmacy, MMDU. Congratulations. Another candidate, Ms. Mamta Devi from Institute of Nursing, MMDU. And the last candidate who has bagged the first position under oral presentation category is Pandey Pratima from National Institute of Pharmacy Education and Research, SAS Nagar. Ladies and gentlemen, we'll have another category. The Young Scientist Award. Ladies and gentlemen, please put your hands together and welcome on stage the winner of the Young Scientist Award, none other than Dr. Sukhbir from College of Pharmacy, Maharishi Markandeshwar, deemed to be university. Congratulations, sir. Next, we have the Best Research Paper Award. And I would like to congratulate Mr. Rajat Goyal and Mr. Vishal Sharma under the said category from Maharishi Markandeshwar College of Pharmacy, Maharishi Markandeshwar, deemed to be University, Mulana, Ambala. Both have gained the title of Best Research Paper Award. Congratulations. and gentlemen, appreciating the young minds, we would like to appreciate the Best Startup Award, which wholly goes to none other than Mr. Himanshu Kumar from College of Pharmacy, Maharishi Markandeshwar, deemed to be university. Please give a big hand to Mr. Himanshu Kumar. Ladies and gentlemen, we have the winners from the Rangoli competition, which was held under World AMR Week. And we have the 3 prizes here, third, second and first. The third prize was bagged by 2 groups. One was with the Dhruv and Paras from PharmD College of Pharmacy. I request them please join us on the stage if they are present. And another third prize was bagged by Jhalak, Faiz, Karanbir and Dipanshu from B Pharmacy College of Pharmacy. Please join us on the stage.
I request Dr. Tarun to please come up on the stage and distribute the prizes. The third prize for Rangoli competition. This is for the information. If anyone want to collect their certificate, they can collect from registration desk. Moving on, the third prize for Rangoli competition, Jhalak, Faiz, Karanbir, Dipanshu. The second prize for Rangoli competition is backed by Pranshu, Simran and Karthik from Farm D. The first prize under Rangoli competition is backed by Divya, Agam and Gursimran from B Pharmacy.
Congratulations. After that, we had another category in this said week, that was quiz competition, and the third prize was backed by 2 teams, one with the Arjun, Aditya and Himanshu, another one by Ashwini, Nikita and Shweta. Both teams are from College of Pharmacy. The second prize was by Sagar, Prashant and Abhishek. First was by Shivam and Jasmine from the said college. These were the participants who were the first prize winners under the category of quiz. Congratulations. Another category was of social media viral star, with the second prize winner of Deepak Malik and Aarti Shukla. Both have bagged the second position under the said category, and the first prize winners are Abhishek Sharma, Divya Kundalas and Princey, under the guidance of Princey ma'am. I'd like to request Princey ma'am to please join us on the stage, as she was the faculty member who was guiding the students under the category of social media viral star. And Princey ma'am as well has bagged the first position under the category of the said social media viral star in the faculty members. Another category was poster making competition, and the second prize was bagged by Simarjeet Kaur from PharmD first year College of Pharmacy, and first prize by Jyoti Jain from PharmD from College of Pharmacy. Thank you so much. If they are present here, please join us on the stage and take their awards. Ladies and gentlemen, all this conference, the work, this all would have been totally impossible without the work, hard work and toilsome midnight oil that the faculty members and the organizers have burned during these 2 days, and the process that had been started before, even before that. We had the members who were also from the College of Nursing, Dr. Vinay Kumari and Miss Manpreet Kaur, who are joining us during these hectic days. We will be beginning with the appreciation of these LOC members. Starting with, I'd like to call upon stage Dr. Dinesh Mehta sir from College of Pharmacy to please join us on this stage. Dr. Reena Das Mehta, Dr. Anuj Malik, Dr. Akash Jain. Dr. Anuj Malik, Dr. Akash Jain, I request Dr. Anuj Malik sir to please join us and take their award. Dr. Anuj Malik.
I request Dr. Akash Jain, Dr. Seema Bansal, please join us on the stage. Dr. Sukhbir Singh. Next, I would like to invite Dr. Neelam. Ma'am, please come on the stage. Dr. Inderjeet Verma. Dr. Tanuj Hooda, please come forward. I request Dr. Jasmine Dr. Yusuf Ansari. Yusuf sir. Dr. Sarita. Dr. Sarita is taken. Dr. Sheikh Karimullah. Sir, please come forward. Dr. Amit Luthar. Dr. Amit Lathar. Sir, please come. Dr. Nidhi Gupta. तुम निकलवा लो वहाँ से awards. No issue, no issue. I request Dr. Sunaina, please come forward. मैम का अनाउंस हो गया? हो गया अनाउंस? Dr. Preetham, please come forward. Dr. Owais. Dr. Priya Diman, please come. Dr. Sandeep Kumar. कैसी बातें कर रहे हो? Dr. Omveer. Dr. Ishrat. Dr. Nagita. Dr. Nagita, please come. Dr. Sandeep Kumar. कीर्ति मैम को बुला लो. बुला लो आप. Miss Kirti Goel. Dr. Tarun Singh. Miss Isha Chawla. Miss Princey Rana. Next, Mr. Vishal. Miss Renu ma'am. Akriti. Okay, Mr. Kashish Goel. Mr. Kashish Wilson, I'm sorry. Miss Garima. Rajat Goel. Mr. Sandeep Rathor, Miss Sapna, Miss Prachi, Dr. Swarna, Dr. Swarna, and last but not the least, Dr. Manisha.
Yes, please put your hands together for Dr. Manisha Bhatia, who was always on toes during these 2 days, maintaining the decorum and as well maintaining all the work which was going on the front stage and as well on the back stage. Kudos to her. And yeah, that too as well. I'd like to announce that Principal Sir, please a request if you could be on the stage for a while.
Now I would like to request all the faculty members to please come forward for a minute. It's really been impossible without the guidance of our esteemed Principal Sir. So just a token of our honor from our side to Sir. I request everyone to join over here, all the faculty members, please come forward. I request Simar ma'am to please come forward on the stage and receive our token of appreciation. Now I would like to request Dr. Dinesh for his vote of thanks, the organizing secretary of this great event. Please give a huge round of applause for Sir.
Good afternoon, everyone. Respected dignitaries, participants, dear students, I am feeling honored to get this opportunity to conclude the DBT sponsored international conference under the aegis of APTI on research ethics, clinical therapeutics, and phytopharmaceutical drug research. It was fascinating and engaging these 2 days, but organizing international conference is a challenging task. So this cannot be possible without great preparations, wise working hands, sports from every corners and blessings. So now it's a time to convey our gratitude to all who have extended their support to make this event successful. At this outset, on behalf of organizing committee, I like to express my profound gratitude to our Honorable Chancellor, Dr. Tasim Kumar Garg for his expected leadership, guidance, and motivation. I extend my gratitude to Maharishi Markandeya Trust for their support in all possible manner to organize this conference. Let me express my deep gratitude to our Honorable Chief Guest, Dr. Y.K. Gupta, President AIMS, Guest of Honor, Dr. V.K. Kapoorji, Emeritus Scientist, Punjab University, Chandigarh, for kindly accepting our invitations and delivering the inaugural address. My sincere thanks to our Vice Chancellor, Dr. Harish Kumar Sharma, who have graced us with his kind words. My sincere thanks to our Dean, Principal, and Chairman of the conference for leading and inspiring us in working toward the goal of conducting this conference. So we are thankful to him for his huge support all the time. So with these words, I would like to thank our distinguished speakers for their excellent presentation and making this conference interesting and meaningful. I would like to thank our resource persons, session chairs, who have given their precious time to make this conference successful. So many thanks to the National Organizing Agency, DBT, for supporting this event and bringing the conference to this level. I'm also thankful to APTI and Haryana Medical Council for their support. So, dear all, so we have received around 550 registrations from both national and international participants and around 50 institutions. So which reflects our team, dedication, and effort. So big round of applause to our dedicated faculty. Due to interruptions in internet, the challenge of registering participants, compiling more than 300 abstracts, coordinating poster and oral presentation, and printing these souvenirs on time is tough for our organizing committee members. So I extend my sincere gratitude to all the members of the registration committee, scientific committee, and souvenir committee for your unwavering dedication and efforts from the beginning until now. So I would like to thank Venue Stage Committee for coordinating inaugural validatory and technical sessions. Your ability to handle every detail with grace and precision made the event a seamless experience. So my many thanks to the media publicity, press committee, and photography, hospitality committee for coordinating the food and refreshment, discipline committee, treasurer, accommodation committee, guide map, and transport committee. Without you guys, these events would not be possible. As you all know, organization of successful generally the result of close cooperations among several institutions. So my sincere thanks go to our joint hand, Maharishi Markandeya Institute of Medical Science Research and College of Nursing and Hotel Management also. I am very thankful to all non-teaching and supporting staff for their support. Not in last, but not least, the heartfelt thanks goes to all student coordinators, volunteers who have done selflessly always behind us for successful organizations of every event and their coordination and sports. So if time is money, then today you have spent million for us.
So thank you to all of you. Thank you very much.
I would like to request all the faculty members to come forward for a photo session. All the faculty members of MMCP, please come forward.
Ladies and gentlemen, with this we conclude our two-day DBT-sponsored international conference on research ethics, clinical therapeutics and phytopharmaceutical drug research, which was organized by College of Pharmacy in association with Institute of Medical Science and Research and College of Nursing. All thanks to the speakers, the resource persons, chair sessions, the guests from across the India and across the borders as well for joining us and leaving the impressing marks on the lives of all the people who attended the conference. Thank you all.
One more request, I request all the volunteers to come forward for a photograph. All the volunteers there inside the room, yes, please come. You have done a commendable job. We really appreciate your efforts. Let's have a group photograph.